Sarah, it doesn't want me to share. So, welcome everybody to the afternoon session. We are continuing with a couple of more emerging scientist presentations. It is my pleasure to introduce our next speaker. She's working here at the UT Health, so she's kind of one of our own. Joana Bianchi. She graduated with Masters of Science in Biomedical Engineering in Universidade de Porto in Portugal, and went to finish her PhD in immunology in Universidade de Lisboa in 2019. She studied the expansion and characterization of donor-specific regulatory T cells, envisaging their clinical translation to use in graft versus host disease prevention. Her background is in engineering, so good background for cytometry, I feel. It's close to my heart. I'm a bioengineer myself. Joanna is very interested in novel unbiased analysis techniques and in expanding scientific knowledge via high throughput data acquisition using flow cytometry. Welcome.
Good afternoon, everyone. First of all, thank you for the introduction and for inviting me to talk here. I know we all just came from lunch, but I'll try my best to keep you all awake. So today I'm going to talk to you, about using flow cytometry to find and characterize cells that are expressing IDO in both the protein and mRNA form. So my group, one of our focuses is on the treatment of traumatic brain injury. And one of the things that we recently found is that the infusion of bone marrow-derived mononuclear cells was able to actually improve the traumatic brain injury outcomes in the long term in both adult and pediatric patients. So as good scientists that we are, the next question was, why is this happening? To answer this, we started by looking a little bit to our previous work with MSCs, where we saw that these cells were able to produce IDO when stimulated with interferon gamma, and that they were also able to suppress the proliferation of activated T cells. A little background on IDO, it is an enzyme that is the limiting reagent in the catabolism of tryptophan, and then it breaks it down into kynurenine in peripheral tissues. So it has been described as having a regulatory capacity because it reduces the tryptophan levels, and then it will cause T cell energy, among other effects that it can have. So our next question was if IDO in the bone marrow cells that we're infusing could be linked to the improvement that we see after the traumatic brain injury and subsequent transplantation. So there are three questions that we wanted to answer with this, which the first one was what subsets were actually producing the IDO. And then by knowing that, we could then ask what's their prevalence in different cell products, such as umbilical cord, peripheral blood, or bone marrow. And then finally, if these subsets could then be linked and used to predict potency in future assays or traced back to our previous clinical trials. So to try to answer this, my first answer, of course, was to try to do it by flow cytometry. So I started by trying to stain IDO protein intracellularly. And what I did is I was working with umbilical cord mononuclear cells because they're easier to obtain for us. And I was plating them either without stimulation or after stimulating for 24 hours with interferon gamma and TNF alpha. After that, I collect them and I stain them intracellularly with IDO1. And you can see that in the three types of samples, I had a positive population for IDO protein, in all of them similar, which was weird to us, but because FMO looked really clean, we thought this staining was working. So based on this, I could then go back and look at umbilical cord samples, bone marrow samples, and peripheral blood samples and try to see the frequency of IDO expressing cells. And you can see that for umbilical cord, it is very diverse. For bone marrow, it was a little bit more stable, and peripheral blood seemed to have no IDO expressing subsets. So knowing that the IDO staining was working, my next step was to design 3 different panels to try to identify the subset that we're looking for. I did this because I'm working on an LSR2, so I only had 8 colors, and I wanted to make sure that I could cover everything that I could. So I first had a mixed panel, which has a little bit of everything, let's say, so I could see if we're looking more for a myeloid or a lymphoid lineage. And then by knowing this, we can then go into our lymphoid panel, which would have more subsets within the lymphoid cells, or the mixed myeloid panel, which would have more subsets within the myeloid cells. And I was running the 3 panels at the same time on all of the samples so that I could then go back and analyze all of them. So looking first at the mixed panel, I'm running, again, umbilical cord mononuclear cells. And when I ran FlowSum on these cells that I had plated with stimulation or without stimulation, I could see that there were 2 clusters popping up that were clearly IDO positive here, 1 and 2. And then when I look at the heat map, it seemed that both of them expressed CD15 and CD11BC. So this to me told me that I was probably looking for a myeloid cell subset. So knowing this, then I went back in the same samples and I looked at the myeloid panel that I had acquired. And when I ran t-SNE on it, this is an N of 3 samples with the 3 conditions and stimulated interferon TNF. And then when I look at heat maps by antibody, you can see that there's this big cluster that seem to be expressing a lot of IDO, and that they seem to be intermediate for CD11C and positive for CD15. So with knowing this, I went back to conventional gating, and I wanted to identify these cells without having to do all of these fancy analysis techniques, because the idea is to move this to the clinic as a criteria for our clinical trials. So what I did was I get it on CD15 cells, and then I get it on negative CD11C and low CD11C, because it seemed to be what I was seeing in the heat maps. And when I go back to my t-SNE plots, I see that I was actually being able to separate out this IDO expressing population really well. And again, here, when I look at IDO, it seemed that this gating strategy was the correct one for isolating these cells without needing further intracellular staining. So the problem started when I decided to run our thawed bone marrow samples. I started by running a test where I froze. I ran a sample fresh and then froze it and thawed it. And apparently these CD15 positive cells were gone when I thawed them, and the IDO positive cells as well, which kind of made sense with the subsets that I thought we were talking about, but that made our life harder. And then when I went back to umbilical cord and bone marrow samples, I validated that this was the case. The thing is, when I ran this sample on qPCR, because I had frozen pellets from this, I saw that, first of all, the IDO mRNA was only increased when I stimulated the cells with interferon, which is different from what I was seeing in the intercellular staining. And also, the thawed samples have had even more mRNA than the fresh ones. So to me, this raised a lot of flags to us, because we were like, is it different subsets? What's going on? Because we're seeing different things in protein and mRNA. So of course, as a good cytometry lover, the answer is let's do mRNA by flow. So we digged a little and we found this prime flow assay where we can actually label mRNA using flow cytometry techniques. It's a 2-day protocol. It's long, but it works. So what we do is I also ran probes for beta-2M as a control to see if the amplification worked. And you can see here that I'm seeing beta-2M on my control, and then my unstimulated cell sample has no IDO1 mRNA, while the stimulated with interferon 1 has IDO mRNA. And this lined up really well with our qPCR results. So I also had another second red flag here, although because when I look at CD11C, if you remember, this was my criteria for gating on the protein expressing cells. And you see here protein, they have CD11C negative. And then when I look at the mRNA expressing ones, they're CD11C positive. So the next answer for me for this was I need a bigger panel, and I need to do protein and mRNA at the same time so that I know if they're the same cells or if they're not the same cells, what is going on here. So I went back to the drawing board and I designed another panel bigger where I had my main antibodies of interest, and I had both the protein and mRNA included in the same panel. I had to move the IDO antibody into PE instead of Alexa Fluor 647, which is where I had it because I was doing mRNA there. And then I tested the panel, and I started again with umbilical cord blood, And you can see here that in my unstimulated cells, I have a little bit of IDO protein positive and a little bit of IDO mRNA positive. But then when I do stimulate them, you see clearly that there's a big population of cells expressing both protein and both mRNA. And they're all CD14 positive, the ones that are expressing IDO in any form. To just make sure that everything was right, I also ran FMOs, and you can see that there's a lot of background for the protein, but the positives clearly are positives. And the mRNA, the FMO for mRNA is really, really clean, so it's not hard to distinguish. The other thing is I went back and I looked again at CD15, which was my initial criteria, and you can see that the IDO expressing cells here, they're not CD15 positive. So to me, this just confirmed that the IDO antibody that I was using at first was just not right. So now going to this new data that all seem to line up, I went back and tried to find out a new gating strategy, and I saw that the CD14 positive CD11C cells, if I get it on them, I would get over 90% of the IDO, double IDO expressing cells. And if I did it the other way around by getting first an IDO positive for protein and mRNA, I would get mostly CD14, CD11C cells, which tells me this is the population that I'm looking for in our samples. And the good part is that because I ran all of those panels in the cells, although I don't trust the IDO staining, I had the 14, I had the 11, so I can go back and check prevalence of the subset in all of them. And I was able to do so. You can see the interesting part here for me is that the number of frequency of monocytes is increasing when I do stimulate it. I don't know if it's increasing in numbers or other cells are dying off because of the stimulation, but this doesn't seem to happen in peripheral blood, which I don't have answers for this yet. It's just something that I thought was interesting to look at. And then when I also go back to my Disney plots for the myeloid panel, this cluster just seemed to be staring at me the whole time. I just missed it because you see here, these are the interferon samples. They all are together in one cluster. They're CD14 positive, they're CD11C positive. But because they didn't line up with my IDO at the moment, I ignored it and I wasn't looking for it. So this also will tell us a lot more. I was digging through the data. There are more markers that I'm going to look at, but this is just a little teaser of it. So just to make sure that these CD14 cells could be promising for our clinical data, I wanted to see if they could be suppressive. So what I did is the same cells that I was plating with and without stimulation, I separated the CD14 positive cells at the end. So then I co-cultured them with PBMCs that were stimulated with beads. And you can see here, this is them proliferating alone, like here, like these little peaks. And then when I add the bulk samples, there's not a lot of suppression of the proliferation. But then when I add the CD14 cells that were separated from the bulk, you see that the ones that were stimulated previously with interferon are able to really suppress the proliferation of the T cells really well, especially considering we're talking about monocytes. So this is the division index calculation for 2 different experiments that I ran. I'm going to run more. But overall, I have 2 types of conclusions for you today. The first part is please cross-validate your results because we could have stayed with a CD15 subset and that was wrong. Also check the specificity of new amps you're testing, new antibodies. And then this is just a personal one that to me, it's always better to have more colors and see more things in the same panel because it makes you not have so many doubts. And then finally, I was able to develop this really cool new mRNA labeling technique using flow cytometry. And we're really excited about being able to use it in other things. And then scientifically speaking, I was able to see that the CD14 positive monocytes were the main IDO producing subset. So now we can go back to clinical data and to future data and look just at this subset, which is much easier to label. It's not a 2-day protocol. And then that these activated monocytes, which would be actually described as pro-inflammatory, seem to be suppressive in this setting. We're going to test more settings, but that's what we have for now. So to conclude, I would like to thank everyone in my lab, especially Scott, for always letting me try these new expensive things. We both get really excited about flow, so it's good. And that's it. I'm open for questions. Fresh. Mm-hmm. Yeah. Yeah, no. So in the first batch of data that I was showing you, where I was looking at the CD15, all the samples we collected them fresh. Sometimes the bone marrow was shipped to us overnight because we bought it, and then I plated them for 24 hours and then collect and ran the panels. But I never let them wait around for very long. Also, yes, the fact that they're gone, that's what I thought was happening when I saw them and the 15 positive cells are not there. It actually made sense with the subset that I was looking at. It's just not what I was looking for.
Do you have any tips for anybody else who wants to go after?
Don't be scared of it. It's not as intimidating as it sounds. It's just long and tedious, but it works really well. I've run it. Scott's there, he'll know. I was terrified the first time because it's expensive and they were very strict with the protocol and stuff like that. And I was like, what if I'm doing this for 2 days and it does not work? And then it worked and it kept working and it hasn't failed so far. So let me not jinx it, but it seems to work really well. Thank you.
And now we're gonna hear from our last emergent scientist, Jayanta Mondal. He's a Scheisler Foundation Research Fellow, as well as a PhD candidate in the Cancer Biology Program at MD Anderson Cancer Center of UT Health. He is interested in the fields of cancer immunotherapy, elucidating the molecular mechanisms governing metastasis, and deciphering the nuances of cancer dormancy and reactivation. He is working on projects in the Jason Hoos lab that focuses on epigenetic and genetic entities that regulate breast cancer metastasis to the lung and to the brain. Before he joined the grad school, he had studied the role of immune cells in the therapy failure and tumor metabolism at the Harvard Medical School MIT Health Sciences and Technology program. Besides this promising career, he's also an avid debater, and he has been involved in theatrics. He has served as the executive board member in the Model United Nations conferences and has worked as a United Nations gender equity advocate. Welcome.
Thank you so much to FlowTex for having me here. I'm a final year PhD candidate at the Jason Hughes Laboratory, and I'm going to talk about my major thesis project, which is currently under revision. So my talk is titled Neutrophils Conspire with Exhausted T cells to Promote Breast Cancer Lung Metastasis Upon Loss of the Epigenetic Regulator BRD7. It's essential to understand the importance of studying metastasis. Firstly, breast cancer is the most common cause of cancer-related death among women worldwide. Also, more than 90% of cancer-related death is a result of metastasis. 62% of people diagnosed with metastatic breast cancer develop lesions in the lung. Over 70% of breast cancer patients who eventually passed away were diagnosed with lung metastasis. And the median survival for breast cancer patients is only 22 months after being diagnosed for breast cancer lung metastasis, which tells us the immense potential of trying to understand the molecular mechanisms that drive this metastatic cascade. First, it's a bit of background about the invasion metastatic cascade. It's a 6-step multi-process process in which in the first step, it goes through the invasion process, followed by the intravasation process, and then it enters into blood-borne dissemination and enters the circulation. At sites when the cancer cells finds conducive for outgrowth, it extravasates into a distant secondary organ. At this distant secondary organ, the cancer cells enter into a stage of initial survival and dormancy, and only when the situations are conducive for it to outgrow, this imposter-like cancer cells reawaken and colonize the distant secondary organ. So this is a bit of schematic as to how the dissemination of cancer cell leads to dormancy. Dormancy is the phase in which these cancer cells enter into a dynamic equilibrium with the immunity, in which these metastatic initiating cells grow at the equal rate at which they're cleared off by the immune system. And only when these dormant cancer cells are being failed to be cleared by the immune system and surpass the immune surveillance, that's when these disseminated tumor cells colonize the distant secondary organ. Interestingly, at the time of breast cancer diagnosis, tumor cells were found to be disseminated to distant secondary organs in over 60% of cancer patients. About 80% of lung metastasis arose from early disseminated tumor cells. However, the molecular mechanisms by which this dormant breast cancer cells reawaken to undergo metastatic relapse is relatively unknown. So to address these gaps of knowledge, I performed an in vivo loss of function epigenetic screen in which I took over 250 epigenes and transduced them into the 4007 TGL triple negative dormant breast cancer cell line and injected into the mice. Some of the mice developed metastasis. The tumor lesions were isolated, followed by barcode sequencing. And the topmost hit from this epigenetic screen was BRD7, also known as bromodomain 7. So bromodomain 7 is an integral subunit of the PBAF-specific SWI/SNF family of chromatin remodeling complex. And to look further into the SWI/SNF complex, it's an extremely elaborate complex, which is comprising of the BAF, PBAF, and the non-canonical BAF. The BRD7 is a subunit of the PBAF complex. BR7, referring to the BRD7, is specific to only the PBAF subunit and is not present in the BAF or the non-canonical BAF. So we then did a CRISPR-Cas9 knockout of BRD7 in the 4007 TGL mice model, in which we saw that at the 30-minute stage, once the mice were all injected with the cancer cell, you can see through the IVIS luminescence imaging that all the cancer cells are equally injected. However, at the 1-week stage, the cancer cells start becoming dormant, in which they lie like imposters in the lung, and they are hard to detect. However, when we had knocked out BRD7, it led to breast cancer lung metastasis, which showed that BRD7 is a suppressor of lung metastasis, and its absence could lead to metastatic ramifications in the lung. We also quantified the normalized photon flux, in which we could see that upon knocking out BRD7, there was increased lung metastasis, and also there was significant decrease in the person's survival of the mice upon this knockout. We also probed for patient samples to see the clinical relevance. We did find that loss of BRD7 expression correlated with metastasis in patients, and also poor relapse-free survival and also overall survival. Next, to understand the gene expression profile changes, we did RNA sequencing, in which we found that BRD7 loss upregulated oncogenic signaling pathways, like the commonly known hedgehog signaling, EMT transition, PI3-AKT mTOR pathway, and hallmark angiogenesis. However, BRD7 loss also abated tumor immunogenicity. As we can see, the loss of BRD7 downregulated antigen processing and presentation, response to interferon, MHC class 1 expression, and immunogenicity can be defined as low immunogenicity can be defined as loss of antigen presentation and also low response to interferon. And as we can see in the BRD7 knockout, both the groups, there is decrease in expression of genes related to antigen presentation and also decrease in response to interferon expression, which told us that there was something going on with the immune system upon loss of BRD7, which was promoting this lung metastasis. So we did a cytokine profiling through Luminex. We scanned over 40 cytokines, and we found the loss of BRD7 led to upregulation of pro-metastatic cytokines like CXCL10, CCL5, CCL2, and CCL20. These pro-metastatic cytokines have been known to recruit immune cells, which are pro-tumorigenic, to the immune microenvironment. So next, we performed a FACS experiment to understand which particular immune cells were being recruited upon loss of BRD7. This was a brief schematic in which we injected the mice with our cells, either knockout BRD7 or controlled dormant cells. We isolated the lung followed by collagenase digestion and then Percol gradient and RBC lysis, and then we stained with an antibody cocktail of 16 color antibodies. This is a brief schematic of the grading strategy what was used. And immunophenotyping through FACS gave us interesting results in which we found that loss of BRD7 was leading to upregulation of neutrophils, while there was downregulation of monocytic cells, macrophages, and DCs. So this was interesting. So we followed this up with a single-cell RNA sequencing to understand the specific immune compartment which was being modulated upon loss of BRD7. So upon single-cell RNA sequencing, we found that it was the effector T cells which was being upregulated upon loss of BRD7, and also the neutrophil population. The neutrophil population recapitulated our FACS findings, but the new population that came up, which was upregulated, was the effector T cell population. So this was interesting because effector T cells are widely known to suppress tumorigenicity. But over here, we were seeing that loss of BRD7 was increasing, in fact, the effector T cell population. So we thought of breaking this T cell population down into 8 subcompartments. And among the 8 subcompartments of this effector T cell, we found that the naive CD4 positive T cells which was going down. However, the stress response CD4 positive T cells was going up, and the effector CD8 positive T cells. The stress response CD4 positive T cell that you can see here is a recently identified T cell population characterized by upregulation of the heat shock protein factors. And these T cell population, along with effector CD8 positive T cell population, was resulting in increased metastasis to the lung. So we wanted to break this effector CD8 positive T cell function further down into the effector, T effector, and T memory, and CD8 positive exhausted cells. While the T effector and T memory cells were decreasing upon loss of BRD7, we found that the CD8 positive exhausted T cells were increasing upon loss of BRD7, which was leading to breast cancer lung metastasis. We, of course, checked for the different markers of T cell exhaustion, like CTLA4, LAC3, TIGIT, PDCD1, and TOX, and all of them were upregulated upon loss of BRD7. Next, we wanted to therapeutically target our findings of upregulation of neutrophil and exhausted T cell. So first, we use an anti-lysin 6G treatment antibody. So I treated the mice every day with the anti-lysin 6G antibody. This antibody has been shown to deplete neutrophils completely in vivo. We confirmed that through FACS experiment. But however, we can see here that BRD7 knockout treated with the IgG control, there is metastasis. However, when we treat the same mice with the anti-lysin 6G antibody, by which we deplete the neutrophils, we can see complete abrogation of breast cancer lung metastasis, which shows that the lung metastasis is dependent on the presence of neutrophils. Further, we used the civilistat treatment. Civilistat has been known to suppress the neutrophil extracellular traps. Recent literature has suggested that neutrophils excrete this extracellular DNA, which promotes the metastatic cascade. In fact, indeed, like upon treating with civilistat treatment, that also prevented the breast cancer lung metastasis. And thirdly, we wanted to target the exhausted T cell population by using immunotherapeutic interventions. Through single-cell RNA sequencing, we had found an upregulation of LAC3 and CTLA4. So we tried using both the antibody alone and in combination, and we found that while the anti-CTLA4 treatment completely depleted the breast cancer lung metastasis, the anti-LAC3 did not have any effect at all to the metastasis. However, the combination was also as effective as the anti-CTLA4 alone. Also, the person survival in that, we found that all the 10 mice in the group survived up till 50 days in the anti-CTLA4 alone and in the combination treatment regime. Finally, to conclude our findings through a brief graphical abstract, we can say that when BRD7 is present in the PBAF subunit, there is ample recruitment of DCs, NK cells, and monocytes, which maintains the dormancy status of this breast cancer in the lung. However, when BRD7 is absent, there's upregulation of oncogenic signalings like PI3-AKT signaling, HEDGOC signaling, and also downregulation of the MHC class 1 expression and IFN response, also leading to an upregulation of pro-metastatic cytokines like CCL2 and CCL5. There is also increased recruitment of pro-tumorigenic immune populations like neutrophils and exhausted CD8 positive T cells, which leads to the metastatic reawakening of this breast cancer cell in the lung. This could be therapeutically prevented by anti-lysin 6G antibody and the CTLA4 blockade treatment. Finally, I would like to thank my entire lab and the Giancotti lab at the Herbert Irving Columbia University, and my advisory committee members and my sources of funding. Thank you so much. Sure. Yes, so we did see a decrease in macrophages. Yes, upon loss of the gene, of the BRD7 gene, there was decrease in macrophages. Great, thank you so much.
Here, you have to introduce. Where he is. He's around, he's around.
You can slide without going to the menu. Use the cursor.
Yeah.
Okay. This last time I had some trouble with. Okay, good. Thank you.
All right, it's my pleasure to introduce our next speaker, Dr. Bartek, who talked to us earlier yesterday about cytometry part A. He's got an interesting talk today, Deciphering the Spectral Code, How Mathematics of Signal Unmixing Establishes Modern Cytometry. When you read through his bio, it's all very interesting and how it came to be. And I'm interested because it's hopefully what he is doing in his work is going to get us away from subjective gating, if you will, the gating we've all been using for too many years. He's a research professor of computational biology and bioinformatics at the Brindley Biocenter in Purdue University. So you must be just down the hall from Paul Robinson. Different building, okay. Expertise lies in a rapidly evolving field of biological data science, which spans across bioinformatics, biostatistics, and multiomics. He's got quite a number of contributions, but a lot of his research employs applied mathematics through computer science and machine learning to decipher these analyses. And by addressing the issues of automatic classification of phenotype in the presence of incomplete and noisy information, and hopefully get things where we can kind of clean up that information and clean up the data. But this research has had direct impacts on the fields of cytometry, hematology, immunology, and a variety of other areas. Now, I don't know whether I'll still offer condolences or congratulations on becoming the next cytometry editor, but so right now I'll go with congratulations. But I heard last night you gained a new title called the Fork Gifter. So maybe you could also explain that before you jump into your talk. So with that, Bartek.
Thank you very much. Thank you very much. I would like to thank the whole committee for inviting me and allowing me to speak to you. I will defy the expectations a little bit because I will not really talk much about the new stuff. I will talk about very, very old and historic stuff, but I thought that we are at the point that we are moving to those very often inexplainable or unexplainable methods of analysis. And it's sometimes valuable to move back in time and try to understand where all of this came from and why people thought in a particular way about the data analysis. Because if we fully understand their way of thinking and we actually retrace their way of thinking, we will try to basically put ourselves in the shoes of the early pioneers of multiparametric flow cytometry. And when we retrace their way of thinking, it will reveal to us what were the real limitations, what are the limitations now, and also it will allow us to fully not only appreciate in the sense of intellectual appreciation, oh, how cool is all of the stuff that we have right now, but in the sense of understanding, right? Fully understanding how the modern tools operate. And I also thought that this is a nice coda to one of the opening talks that were actually to two opening talks. Joel's talk about the history and David's talk about the history, and also Peter's talk, which I love because he introduced certain metaphors linking the mathematical operations to actual data and how we are really transforming the representation, the data representation of the flow cytometry measurement. And I'm going to build upon his presentation. So those who were unfortunate not to be here when he presented, you will have tough time following what I'm going to say. But anyway, jokes aside, we are all in the lunch coma state. So there's nothing better than having a talk about history and mathematics, or history of mathematics, right? That's going to be fantastic. But history of science is a fascinating subject. And as I said, it's a fascinating subject because studying the past methods and techniques used for processing data really helps us understand how the modern methodology works like. And it kind of demystifies some of the steps, right? Suddenly you will see that, hopefully by the end of this talk, you will see how simple all of that is. It's actually childish what we are doing and what we are trying to accomplish. And so by gaining knowledge about the historical techniques, we can fully appreciate the modern method. So let me start with one color cytometry. And you will see I have a bunch of illustrations of old flow cytometers on my images, on my slides. This is some 17th century implementation. So one color cytometry. Well, you know from the historical talks that were given day before yesterday during the opening day, that obviously the first flow cytometer was devoid of ability to measure any fluorescent signal. But then, end of 60s, independently, two groups, one group in Germany, Professor Goede's group, and the other group in Los Alamos, decided to put a fluorescence detector on what at the time was not even called flow cytometer. In fact, the name flow cytometry came to existence during one of the meetings in Los Alamos, but they didn't call it. They call it pulse fluorospectroscopy or whatever it was, but it was not even called flow cytometry. And they happened to publish their papers within probably months. Wolfgang Goede in Germany and Martin van Dilla in Los Alamos. They are the people who created cytometry as we understand today, right? That was around the time Marvin van Dilla worked at Los Alamos, around the time that Mac Fullwiler worked there too, and obviously he created cell sorting, as you remember from the historical introduction that David gave on day one. So we had one color flow cytometry. Why it was such a big deal when two colors were introduced? And here is the person who did that. That's Mike Locken, who worked in Leonard Herzenberg lab, Stanford. And this is the paper where this all happened. And this is an actual first attempt on compensation, which you see here. These were probably pictures taken from the monitor, I can presume, right? And here is the relevant piece of the paper, which says that even though everything was done not algebraically per se, but in a system of two crossover potentiometers, but he explained how it works, right? He says that the algebraic basis for this technique is simple. The signal in the green channel, F1, right? Since then, we all know that F1 is the green channel. You can cite this paper every time you see that in the description of the FCS files, F1, F2, F3, right? The signal in the green channel is a combination of light coming from fluorescein and rhodamine. Therefore, it could be expressed algebraically in the following way, right? Where R, F, and R are the number of photons emitted by fluorescein and rhodamine. And alpha and beta are the coefficients or efficients for the detection in two colors in this channel. Okay, so what happened is at the same time, people in a very different field had exactly the same issue. And that was the field of remote sensing. Because in the 60s, they started to build remote sensing systems. And their big issue was that they couldn't get enough spatial resolution. But they could get more spectral resolution than spatial resolution. So they had to do unmixing, but of course, signals were overlapping. And the person who actually was the pioneer in developing those systems, happened to be a professor at Purdue, David Landgrip, who passed away a few years ago. He developed the first remote sensing, I would say, computational methodology. At the same time, so there were two groups that got NASA grant to do that. One group at Purdue, the other group at University of Michigan, and this consortium built those tools. And the group at the University of Michigan developed what we now know and understand as spectral unmixing. This is their later report from 75, but the first report which I couldn't find, unfortunately, is the 1971 report, when for the first time they discussed the question of spectral unmixing. If you look at those equations, it looks pretty much the same as what Mike Locken was referring to, and later on, Bruce Bagwell, about whom I'm going to talk about. So their problem was that they were looking at individual pixels, obviously, right, rather than individual cells. So an event was a pixel, but every pixel was associated with a spectral signature, and it had to be unmixed. And the first public report about this is from the conference in Ann Arbor in 1971. So previous reports, which were internal NASA reports, which are now declassified, you can find them. But well, obviously, they are not available in every library. So at the same time, the same problem. And clearly, there was some kind of intellectual crosstalk between those groups. Everybody understood that, okay, we could use linear unmixing models to deal with those spectral signals. So as I said, their problem was slightly different because they were looking at a mixture, but a mixture which was spatially offset, right? So this is what they would like to see is on the left-hand side, and what they saw is on the right-hand side, right? So they saw overlap of all of those signals. They couldn't spatially identify where part of pixels or the subpixel, they call it subpixel resolution, right? Where the signal came from, but they could do unmixing and estimate what is the contribution of different signals. That's essentially what we are doing in flow cytometry, although we don't even have a hope of unmixing spatially where the signal comes from, although now with modern flow cytometry, which sometimes integrates imaging into the mix, we can probably at certain point be able to do that as well. Or actually, in fact, we are able to do that as well right now in certain systems. Okay, so now it's going to be tough. So be prepared, because this is a piece, this is essentially taken verbatim from Bruce Bagwell's paper in 1993. So for all of those years, right, we were talking about 70s, then 80s. It's 93. For all of those years, people were doing two-color flow cytometry. And the reason why they were doing this is because this is the compensation module, which you see on the left-hand side. This is how it was done, right? And of course, there was some algebraic basis for it, but there was no implementation. And the contribution of Bruce Bagwell was to recognize something that, in fact, I guess every engineer would consider obvious at the time, especially those remote sensing guys. But he recognized that, well, we can generalize it to any number of dimensions. Obviously, it's just the size of the matrix. And we could attempt to do it electronically. Remember, these were the times of the early race of very powerful personal computers, right? So you didn't need to have a DeckStation VAX on your desk, which cost several $100,000, right? You could have a PC, and PC could do that. And Bruce recognized this. And again, all of those equations, all of this is from his paper. And he says that the system, which is based on crossing over those potentiometers, right? He says the system has worked well for 2 or 3 colors. Okay, 3 was really maximum you could do, but begins to fail at 4 or more colors. Why? Bruce explains the reason for this color limitation is mainly a practical one, because the number of potentiometer settings or coefficients for performing full spectral overlap compensation is related to the number of chromophores by this function C equals n squared minus n. So for 4 or 5 compensation problem, right, you would need 12 or 20 coefficients. Coefficients meaning 20 knobs that you keep turning and trying to kind of manually find a picture that kind of looks how you would expect. And obviously you cannot do that, right? So that was the main reason why for all of those years, 2 colors were the standard and 3 colors was like, whoa, somebody is, hardcore, right? And nobody attempted more. Now, eventually the computers emerged, right? And here is another early flow cytometer from the early computer era. And this is what Bruce Bagwell gave us, right? It's almost looks identical to what Peter presented on the first day. So he had those diagrams in which he is showing that, oh, the signal is crossing over from one channel to another channel, and we can write the same equations in the matrix form. And Peter explained how this matrix format should be understood, right? That this is really the proportion of the number of photons, and the abundance in Bruce's notation was S, and the result in Bruce's notation was O. I actually hate his notation because always, if you see O, it looks like 0, so I'm not going to be using his notation anymore. But then he wrote this equation in the middle of his paper that O equals K times S plus A. So A is autofluorescence. We can ignore it because actually autofluorescence could be built into the spectral signature of every of the dyes. So we don't care about it. It's as the additive thing, but it's simply K times S, right? Again, that was introduced on the first day. So with this observation, one could immediately move to trying to unmix this electronically. But here I'm going to, I borrowed a few slides from my very old presentation, which I actually delivered at this meeting in 2019, which was going in details about spectral unmixing. So I borrowed 3 slides from it just to revisit the denotation again, because I think it's very important to demystify the notation. So the way how you should think about spectral mixing is that it really conveys the proportions in which signal is distributed between the detectors. So if you have 2 types of photons, quote unquote, or some kind of measurables, and you want to have them all fall into a particular detector, but they don't, right? For some reason, doesn't matter what's the reason, only 80% of them go where they should go, and 20% go somewhere else. You can encode it in this notation as a matrix with a column which says 0.8 at the top, 0.2 in the second row, right? And conversely for the other one. That's what mixing matrix really conveys. Simply the proportional distribution of your signal in the detector, right? Okay, so we have that. Now, if we, and this is another of those slides which I borrowed from my last talk from 2019. So if we have the ground truth, we start with 100 photons and we put them through this dichroic mirror, but they are not going all into the red detector. They are spread, right? We are going to see the following situation. We have 100 photons, this is ground truth, and 50 green photons. But after optical separation, we have in one channel 95 photons, in the other channel 55 photons, right? So it's not 150. Not only that, they are not pure, right? In the sense that in one channel where it's supposed to be only red, we have 80 green and 80 red and 15 green, and in the other one, we have 35 green and 20 red. The notation for it, again, we have a column vector, right, for abundance. Abundance is in this case ground truth, and we have this mixing matrix. We need to introduce one more thing. That's the result. So it's another column vector. Again, I'm just recapitulating what Peter said on the first day, and we have the result vector. So how we put this all together? Very, very simple. It's a multiplication of vector by a matrix. So to calculate the result, right, to predict what is going to be your outcome, if you have this type of system with this type of efficiency of separation, you multiply the corresponding components of the first row, and then you get 95, and then you multiply the the corresponding components of the second row, right? And you get 55. That's your result. So this is where your result comes from, simply mixing operation. Now, you can represent it in this format, or you can represent it in another format where you flip those matrices on the side. It doesn't matter, but I kept the slide because I want you to remember the operation of transposition, in which you basically take this box with numbers and you flip it on the side, right? That's the transposition. We'll need this later. And that's the letter T, which you see here, okay? Now, can we invert it? And again, Peter told us that we can, and this is the way how we do it. We basically are trying to find another matrix which, if multiplied by the matrix that we mix this data with, it's going to give us a unity matrix, which we write this way. It's a matrix with 1 on the diagonal. This is matrix representation of basically number 1 in just basic algebra, right? And there is such a matrix, and we can identify it, we can find it, or a computer can find it for us. That's the matrix, that's the result, which means that if we want to unmix what we mixed or compensate, right, we just multiply the R vector, the result vector, by this matrix. We should be back to where we were. And indeed, this is how Bruce Bagwell proposed we do that, right? So he said, okay, calculate the invert matrix and multiply by the data, and that's how the flow cytometry should do compensation from now on. This is what's in his paper. However, that's not how cytometry really adopted this, which is interesting. And let me explain what's the difference. So in his paper, he said, well, take the results, take the mixing matrix, which you can estimate using single color controls, right? The mixing matrix is always formatted in such a way that columns sum to 1, right? Always it's column-wise normalized to 1. Calculate the inversion of the matrix, multiply it by the result of your observation, and then you will have the ground truth. And as you remember in the example, we had 100 red photons, 50 green photons, so we get it back. But that's not how cytometries do it. And you remember it probably, again, from one of the few talks at the beginning, where the mixing matrices or spillover matrices were shown. They had all 1 on the diagonal, right? So you could be asking, wait a minute, this is not what's in Bruce Bagwell's paper. Why the hell we have 1 on the diagonal of every compensation matrix? Okay, because this is how really people operate. They take the value in the mixing matrix and column-wise, they divide it by the biggest value there is. It's called diagonal normalization, right? So 0.8 divided by 0.8 is 1. 0.2 divided by 0.8 is 0.21. This is what really cytometries do. So now you have the explanation why your matrices are actually having 1s on the diagonal, but most of the software that you don't really see, they use matrices which use this other formatting that came from spectral imaging, and basically every other spectroscopy field assumes that mixing matrices should normalize to 1. Well, does it matter? It does, because look at the result. The result is different. If you multiply the inversion of this matrix, which is normalized on the diagonal, by the result of your measurement, you don't get ground truth. You don't get 150. You get 80-35. What the hell is 80-35, you may ask? Well, 80-35 is the subset of the correct photons in the correct location. You see, in the red channel, we get 95 photons. In reality, we have 100 red photons in this example. But in the red channel, 80 of them are actually red. And in the other one, we have 50 green photons. But in the green channel, so to speak, in the dedicated detector, only 35 are actually green. That's what this renormalization gives you. It gives you the result, which is subset of what you actually measured. In fact, in one of the editions of Howard Shapiro's cytometry handbook, he recognized this, that the results are different, and came up with a name for it, which didn't stick, although for some time, Howard was adamant about underlying the point that these are not the same results. And he called this subtractive compensation, and he called the actual, what Bruce Bagwell proposed, additive compensation. Fortunately, nobody picked it up because it doesn't make sense. It's just a different way of representing the data. It's different information. Compensation is compensation. Compensation is actually unmixing under a different name, so it didn't matter. But that is the difference. This is what you actually do if you multiply by the inversion of the matrix, which has one on the diagonal, what you see as a compensation matrix. If you actually wanted to unmix, you would multiply by the renormalized version of this matrix. Okay. Another thing that Peter said, which was excellent, was that in reality, you can think about it as a bunch of geometric transformations. So we have the ground truth, the signal which we want to see. The signal which we measure kind of looks like that, like the image in the middle. It's deformed. It's deformed in a very particular way. It's skewed and it's rotated. And then we try to do compensation, which means we want to go back to the original form. Okay, so I thought, all right, let's take this analogy a little bit further. How would you think about it in the real life? And I will come back to this. And there is actually a mathematical way of thinking about this whole process. And this transformation in mathematics is called polar decomposition. So it's a form of transformation in which you take a matrix, like the mixing matrix, or its inversion, compensation matrix, and you decompose it into two steps. You stretch the values, and you rotate the values, or you rotate the values first, and you stretch the values later. So it means that we can think about compensation along the tools given to us by polar decomposition. It's actually a very important tool for engineers, because they can describe any deformation this way, right? So let's use this analogy, but use this analogy in something real, something that you can imagine. Imagine that you have a piece of stretchy fabric, right? Which is interwoven this way. And first you stretch the fabric, or you write a text on it, right? And then somebody takes it, stretches it, and rotates it. And obviously, the text is not going to be very readable, right? So what's compensation? Compensation is nothing else. It's simply re-rotating this stretchy fabric and repositioning into original position and letting it contract. And this could be described by those two matrices, right, which are equivalent to the compensation matrix. So the compensation matrix, as we saw with polar decomposition, can be broken into those two steps. So instead of having one matrix, which in this graph is shown as A because of this definition that A equals U times P, but that's M, that's your mixing matrix. You can think about it as rotation, that's U, and then P, which is skew, right? This is what mixing is. And obviously the reverse process would be compensation or unmixing. So let's do that. Let's ask ourselves, can we really do that? Can we actually do some darn math and calculate this? Well, yes. So let's express compensation, which is the inversion of mixing, this way. So we take a mixing matrix, we invert it, right? We're going to be calling it C now as a compensation matrix. These are the values. We take it, we let the computer decompose it into two pieces in polar decomposition. This is what we get. It looks scary, but it's actually nothing weird. I mean, the numbers are long because, you know, I started for the purpose of demonstration with nice round numbers. But of course, after decomposition, they are going to be weird. But if you think about it, it's simple. The first matrix is the rotation matrix. The second matrix is the skew matrix. Now, the rotation matrix has a very particular form in algebra. You can look it up, this explanation in Wikipedia. We can write it as a matrix of trigonometric functions of the rotation angle. So it just happens that this example that I came up with, which was 0.802, right? This very simple compensation matrix is equivalent to rotating our data by 3.8 degrees. Then this is what it is, right? We are rotating our data by 3.8 degrees. And we know that, but well, it happens to be 0.06 radians, but for radian challenged people, it's 3.8 degrees. Okay, and we know that because those numbers here, we can do arc cosine, arc sine, and calculate this, right? So this is really what the rotation is. And then there is a skew, which is represented by this other matrix. Okay, so if I took the actual data, this is my ground truth, right? And I had a data which kind of looked like that, and I had my ground truth. After measurement, this is what I would see on the screen, right? It would be skewed and rotated. And now I can compensate, and by compensating, I mean I can deskew, right? But it's rotated still, it's wrong, right? But it's deskewed. Or I can derotate, but the skew will remain, right? So now it's rotated by 3.8 degrees, but it's skewed. It's kind of compressed. Or I can do one and the other. If I do both, then my data comes back. All right, so these are all the transformations, right? So what's compensation? Compensation is taking your fabric, which was stretched and rotated, on which you wrote a text, to compress itself and rotate it back so you can get the information back, right? And these are all the transformations. So we have the original data, the unmixed data, the data which was deskewed but not rotated, and the data which was rotated but not deskewed, right? Very, very simple. This is all there is to it. And if you ask me, well, this is all great, but you have only 2 colors. How do you do it in 3, right? Well, there is a metaphor for that too. That's the deformation of a 3D object in 3 dimensions, right? So you start with the cube, and you can deform the cube by rotating it and stretching in any arbitrary direction. That's what measurement does to your data. And the process of going back, deskewing and rotating it back, that's compensation. And that's, in fact, also unmixing. The only problem with this metaphor in higher number of dimensions is that those angles of rotation, they are called Euler angles, right? And those angles grow quite dramatically. So for 5-dimensional problem, you will have 10 angles, right? For 2-dimensional problem, you have only 1 rotation. But for 5-dimensional problem, it's rotation across 10 different angles in the 10-dimensional sort of system. You may think about, well, system is 5-dimensional, but there are 10 angles, possible angles of rotation. So it's not particularly great metaphor, because it gets from, oh, let's stretch a piece of fabric, to, oh, let's imagine 5-dimensional space with 10 angles of rotation, right? Well, okay, so it breaks down. But it remains true that this is all we are doing to our data. We are stretching and rotating. That's it. Now, because of that, we can say, all right, so we are going to follow what Bruce Bagwell presented in this paper. We are changing his notation. There is no O, there is a result. So R equals the mixing matrix times the vector of abundances plus some uncertainty. And I'm not going to talk today, even though I originally planned, but I thought I'm not going to fit this into time. I'm not going to talk about the N here, which is the noise or the level of uncertainty, because this is a wide, big elephant in the room. All our assumptions so far are that N is Gaussian distributed with the mean of 0, and it's not. And that's why this process never actually works perfectly. And this is why you have the spreading error. But let's set it aside. That's a subject for another discussion. What I want to focus on now is that, okay, if this could be done for any number of dimensions, well, yeah, let's just go, with a higher number of dimensions, or even spectral. We have more detectors than sources. And the only difference, and again, I'm basically repeating what Peter said on the first day, is that we replace the process of inversion of the matrix with so-called pseudo-inversion, which is a little bit more complicated. It has additional step. It's basically inversion of 2 matrix transposed multiplied by matrix. Well, inverted and then multiplied by the transpose matrix again, the same thing, right? It gets you the correct result at the end. And as I said, if we did this for a multidimensional situation, right, with 4 channels, even we have two objects or 2 colors that we measure, but we have 4 detectors, we could first mix it, and then we could unmix it, and we should be back exactly where we were. Everything is fine. However, and that's where the problem starts. What if we try to do it very fast? And you would say, well, the computers are fast. You just did that. I mean, what is the big deal? But no. When I'm saying fast, let's do it in a sorter, right? And in late 90s or mid 90s and then late 90s, people started to think about developing super fast sorters. And those super fast sorters developed at the time were supposedly sorters which sort across, I mean, they take information from multiple channels, right? And the prime example of the high tech of this era was cytomation MoFlow, right? 70,000 events per second, right? So we take cytomation MoFlow and we can ask ourselves how they did that. Did they do those operations, those mathematical operations? They don't seem complicated, right? I mean, you connect computer to the system and it should work in real time. Well, here is the problem. And obviously the picture of MoFlow inside. Every operation we discussed so far is done on the it's a linear algebra operation. And it's assumed that the data is represented in the linear space, right? Everything is linear. You know that that's not true, right? And what if somebody needs to do it in real time, taking under the consideration that it's not really linear data? Well, that was a major challenge. And cytomation people, actually, they were a bunch of geniuses. They solved this problem. They actually published this, well, they didn't publish anything. They filed a patent, and obviously patent is public, so you can look into the patent. The patent was only issued in 2006. The technology was obviously introduced in MoFlow in like mid 1990s, but it takes time for the patent to be issued. Description of all the details are not even in the patent. They are in the original attachment to the provisional, which you can dig out because those archives are also public, right? This is all publicly available information. And it's typeset in a way that actually makes your head hurts because they didn't typeset all the mathematics normally you would expect to be typeset. It's basically written in a text editor, like a normal text. So it's horrible. I don't know if they did that on purpose so nobody understands what they did or that was just came with territory. I don't know. I haven't been born yet. You know, I'm very young. I don't remember. But that's the first page of the patent. So what those guys did, they thought, okay, the major problem is we have log amplifiers. We cannot switch back and forth between linear and log representation of the data because we need to do it very fast, and we do it very fast by doing it, well, we do it very fast, we do it using DSP chip. And there is a certain time and number of operations we can do on a DSP chip. During the time that cell presents itself, is measured, the DSP chip have to calculate everything, and this has to actually affect how the data is sorted, right? So we have very limited amount of time. Everything that Bruce Bagwell proposed is done in a linear space. Everything that Mike Locken described as the algebraic basis of the operation is linear, right? And we're in the log space. What are we going to do? And their solution was, well, let's try to find an approximation. So instead of doing this, actually doing this, they said, is there another operation which gives us precisely the same result and does not require what I just described to you, right? But it's also stretching of a fabric and rotating of a fabric. Let's see, is there another one? Well, it occurs that there is. There is a very simple mathematical trick, which actually it's nowhere named in the patent. There is only result, which you have to reverse engineer. But what they did is they said, and actually this animation in the corner for those who are mathematically inclined reveals the game. What they do, they start with a very simplified version of the compensation problem. So this is one channel, right? And they start with kind of a thinking along the compensation matrix, which has one on the diagonal. So they basically say the voltage in linear space, which is compensated, is this actual measured voltage, and that's why it's L in the linear space from detector number 1 minus some a percentage of the signal from detector 2, right? This is, I'm talking about the two color problem. Of course, they did that in a multicolor, but okay, let's stay with 2 because it's easy to understand. So that's the problem. But they are in the log space because they have log amplifiers in their system, like all the PMT-based sorters, they all use log amplifiers, right? So in reality, if we have log amplifiers, this is what we are measuring. So the compensation should be written this way, right? That the compensated voltage should be logarithm of exponent e to v1, right? Minus some compensation factor times e or exponential function to v2, right? This is what they really want to do. But how are you going to do that fast? This is actually quite complex operation if you want to do all of this on the DSP chip, right? Logarithmic exponents. So they said, okay, let's use so-called Taylor expansion function. What's Taylor expansion? It's a way you're approximating a function at a given point with the infinite series. Okay, I know, I lost you guys. You probably don't want to listen to this anymore. But yeah, let me stab you again. This is what this is what Taylor series is. So you approximate this term, right, with this infinite term. There are only 6 terms here, but this is an infinite sort of. There is infinite number of terms. The important thing is that if your sort of compensation factor is small, you can cut out those additional terms and just take the first term of the expansion of Taylor series. And this is written here, right? So this compensated voltage from log amplifiers in MoFlow in 1995, right? Ancient history would be this term minus exponent or exponential function of V2 minus V1. Obviously, we can convert that. This is obviously just V1, right? Because the exponent and logarithm, this is natural logarithm, not the logarithm base 10. This disappears. So the whole compensation process, if you have log amplifiers, is simplified to this term. Is the original voltage from the log amplifier minus some fraction, right? And then exponent to the power of V2 minus V1. So you need to do the exponential function only once, and you don't need to do any logarithms, right? You can do it faster on a DSP chip. Let's check how close this approximation is. Let's check how close are all the sorts that people did from 1995 till, whatever, because after MoFlow was taken over by Beckman-Coulter, the system was still sold under a different name. And let's see how good those sorts are, right? We can retrospectively check. So we are going to apply the rotation function and see how good is the rotation given this approximation, because we already established that compensation and unmixing is nothing else but skew and rotation. We're not going to check skew. Let's just check rotation because we don't have a lot of time. Okay, so we are going to rotate the data by arbitrary number, but I just picked for the fun of it pi divided by 20 as my angle of rotation. I don't know why. I don't remember why I picked that. Doesn't matter. So that's the rotation matrix, which comes with it, right? It doesn't look like compensation matrix. It has minus in it because, as you remember, compensation matrix is this thing multiplied by the skew matrix. This is part of compensation. This is not whole compensation. And this is my data that I want to rotate. So you will see it forms a square, okay? And my rotation matrix, now I want to express it in the way how typically this would be expressed with one on the diagonal. So I put the scaling kind of scaling of this aside, and I'm going just to be looking at pure rotational properties of it. So after I do the operation, it all looks scary, but it's pretty much follows what Peter taught you on the first day, right? You multiply stuff, and these are the positions X and Y of all the points after the rotation, right? So this was X and Y of point 1, X and Y of point 2, and so on and so forth. Now, after rotation, that's X and Y, X and Y, X and Y, right? We have 4 points. Okay, so that's how this rotation looks like. So if we had exact solution, infinite number of time, if we collected the data and then use software like FCS Express, FlowJo, anything, after the collection is done, and we wanted to do only half of the compensation, just rotating, not skewing, that would be the result, right? That's the data, that's the rotated data. Now, let's use MoFlow. Okay, that's going to be lots and lots of tedious algebra. So we start with the thing which we ended up before. We put it to a slightly different form. This is just algebraic modification because we want to plot this in the linear space. So the solution was developed for the logarithmic situation, but I cannot think in a logarithmic way, right? So I want to plot the thing on the actual plot. So I need to go back to the linear form. So I put logarithms everywhere on everything. And I just, for the fun of it, you will see why I substitute A with some particular term. This actually doesn't matter. What matters is that at the end, after all of those, and after removing the logarithm on both sides, when I went from the logarithmic form, right, VC is the voltage from the log amplifier. This is VLC, which is linearized version of this voltage, right? That would be my compensation equation for this abbreviated superfast compensation in the linear space. So if I have this equation, I can actually calculate rotation of X and Y values, right? If all the points are rotated. So I will have those compensation factors, and this doesn't look like normal matrix inversion because it's not anymore. I have the X and Y position of all the points. I calculate those factors from the rotation matrix, right, which is part of the compensation matrix. So I have this calculated. It looks all complex, but it's just putting numbers into different equations. It's just simple, right? Once you do that, you can see the results. So I put those numbers there. I reformulated it, by the way, in this funny way, because it happens that if you do this operation, this angle of rotation, if you express it this way, is expressed in those equations as a tangent. So you can actually see what this rotation really does. So now we are doing approximate rotation by P20 radians, right? So this is our input. And after we plug in all of those numbers into those equations which we had here, right? We plug them in here with the C which we calculated here. We get X and Y, right? And we can plot those X and Y. So this is what the output of the sorter would be. How good this is? Let's compare. This is, by the way, a MoFlow which was decommissioned in one of the labs and was on sale, I think on eBay or somewhere for next to nothing. So I thought that it would be nice to put it there. So this is the exact solution. This is the solution which they developed to make it actually work on a sorter. And that's the difference between one and the other. So you see, it's almost exact. So for all of those years, when people were sorting on MoFlow, they didn't realize that their population was just a little bit shifted. But it was good enough, right? And it was operating extremely, extremely fast. And that's how pretty much the same type of trickery, this is, as I said, from the MoFlow patent, but the same type of trickery was implemented in all the traditional multi-parametric sorters. So you probably didn't realize that what you learned about compensation and unmixing, all of those matrices, and you felt like, okay, we actually know how those machines work. No, you didn't know. They didn't work the way how you were taught because it would be too slow, right? Nobody would be able to implement it. They actually work in a very different way. They gave you only approximate solution, right? And this is the solution which they gave you. Okay, so what did we learn today? That measuring more than one signals always involve a signal crosstalk, right? That's, I hope I convinced you that that was the case, but my talk was only building on the previous presentations who were explaining how the signal analysis in flow cytometry works. Joel's, David's, and Peter's, right? You take all of those talks together with mine, and I think you have a big picture. Early in the flow cytometry development, the limitation and so-called manual compensation or manual correction was due to the fact that everything was done through electronics. And you couldn't really realistically do it in electronics if you had more than 3 channels, right? Then, with the development of fast electronics and computers, people thought, why don't we do it in software? And you could do that in software very early on with mini computers and then microcomputers, but you couldn't do it in real time, right? So you could collect the data and do compensation afterwards and then do visualization, but you couldn't do it in real time to sort. The first people who solved this problem were the people who work with Geir van den Eng at Cytomation and his subsequent companies. They thought, okay, why don't we use DSP chip to build it into the machine? And that would give us super fast sorting opportunity. But unfortunately, the chip is limited. It cannot do so many mathematical operations. So instead of calculating the actual compensation, we are going to calculate approximation of the compensation. And obviously, that was publicly released as a patent. If people read patents, they would know. Most people don't, especially they don't read early on sort of attachments to the patents which are not found through Google, right? You have to go to actually US Patent Office and dig this document out. That is how it was done. So the process of unmixing, which they were struggling, we are struggling till now, it's simply a process of a linear transformation in which data is skewed and rotated. And all we try to do is de-skew and de-rotate the data. As I said, there is a big elephant in the room. The process is not linear, even though we pretend it is. What it means in this metaphor? It means that we think about skew and rotation like all of this fabric would be stretched in the same way, but it's not. If you stretch it more, right, it actually grows, the stretch kind of grows exponentially. So there is a point of stretching in which you are starting to tear the fabric. You can think about it this way. Therefore, our compensation or unmixing will never, ever give us the ground truth. It can give us the best estimation of the ground truth, or it can give us lousy estimation of the ground truth if we assume that the signal is Gaussian. If we don't assume that the signal is Gaussian, if we assume that it has another distribution, the result is going to be better, but it's still not going to be exactly the ground truth, right? We lose some information no matter what. And all of this in early days, as I said, has been implemented by people smarter than us using DSP chips. And now, obviously, it's a different world. People do it differently, but that's where all of those limitations came from. And the other lesson I think that we all learned is that there is a superficial version of explanation how technology works, and there is actually how really technology works. And very often these are kind of disconnected, right? People don't realize that the textbook version is very often a model which cannot be practically implemented by anybody. Nobody is able to build what we are presenting to our students. So we look for shortcuts. And I hope that this talk explained one of the, in my opinion, most fascinating shortcuts in the history of flow cytometry. So I thank you very much, and I part with you presenting a picture of the Bigfoot sorter. And once again, I appeal to you to support cytometry and submit papers. Thank you very much. Yes, but instead of doing a transformation on every term, right, and then doing operation and then transforming it back, you subtract in the log space and then you do transformation once. So that was the game. There were some other tricks which they implemented, which actually this change set up the ability to do, and I didn't expand on those. But the patent, which is publicly available, is fascinating because they actually attach the assembler code for the DSP chip and every step, and why they were trying to like carve every nanosecond out of this darn thing, because they wanted to gain, a few more hundreds of events more in sorting. It's absolutely fascinating how they did that. The level of optimization is mind-boggling. Nobody writes software like that anymore, right? I mean, we have a software to, I don't know, make a beep, and the software is 2 gigabytes, right, on our computer. Those guys wrote software to do all this stuff. They packaged it onto DSP chip, and they tried to, as I said, gain any nanosecond they could. That's why this technology was so powerful.
Comment, if I may. I will never argue with you about math, for starters, but I'm hoping that as you wrap your head and keep going with cytometry part A, every now and then we see a from the desk of the editor. I would like you to keep some of the history of flow going. With every new machine coming out, it seems like more and more becomes automated, which is good. Right. But at the same time, this is the kind of history that we don't need to see fading in the rearview mirror.
Yeah, I agree that it's valuable to keep all of this, because it's not only the ancient history, right? Now we have those extremely sophisticated automated systems. And half of the time, if you ask people what those systems really do. Yeah. So, 20 years from now, somebody will come here, maybe it's going to be me, and will tell you what your system actually did 20 years ago. And you would be shocked, like, oh, really? They didn't tell me that. I signed a license, I didn't know, and that's kind of a funny example of those constraints.
Anything else? Ryan.
So that's a great question. Sadly, well, I'm not aware of those efforts, right? Not in the academic space. I hope that this is what is happening in places like BD. Probably Peter, after hours, he's trying to optimize this square root transformation so it runs 1 nanosecond faster. And this is actually a great example, because I remember I saw this video explaining why the code for the simple transformation, if you actually see the algorithm, is so mind-bogglingly complicated. And they are essentially shaving nanoseconds out of every cycle by doing this. So I'm hoping that flow cytometry companies are on the same level of engineering now as they were years ago, and they are doing exactly that. Because obviously the problems are more complex, the electronics is much faster, but still it pays to invest in those optimization procedures. So even though I'm not personally aware of this work, that's simply because I'm on the academic side, right? But I hope that that's exactly the case. So when we go to spectral sorting, the problem is of a different nature, right? Because we cannot do linear transformations anymore. I mean, we could, or we could do approximations of those, as those guys did years ago. But in reality, everybody is expecting to be able to gauge the population in some manifold learning space, whether it's PHYT or UMAP or something like this. So now we are talking about really a lot of computation happening. And if you try to do it in real time, that would be almost impossible. I remember when UMAP came up first time, and Anna may confirm that everything worked pretty much at the speed that you could start the process, go and get a coffee, and then come back, and then think about it, and then it calculated what you wanted to calculate. And then some people discovered that, oh, by the way, all these algorithms were not optimized to do anything, and I was one of them. That if you tweak them, then you actually don't have enough time to get a coffee, but you have enough time to scroll through your email, right? So that was a big progress. But now we are talking about doing it in real time, right? So to do it in real time, you have to throw a really fast silicon on it to be able to engage in this. The additional problem, which is, and this is beyond all the technical matters that I covered in my talk, is the deeper philosophical problem with the interpretation of this data, right? And I keep repeating this. You probably, if you sat next to me during lunch, during this meeting, you have heard me saying this, that the fundamental assumption when we do manifold learning is that the data is continuous and well spaced and equally spaced in the space, and it's well sampled, right? But we do it to show clusters of cells. So we are violating the fundamental assumption upon which we build our data analysis. And this gives me a little bit of a problem, right? Because we don't know to which extent some of the visualizations which you have are like the anecdote says, all the models are incorrect, but some of them are useful, right? Those results are useful, but to which extent they represent correct mathematical representation of the underlying biological data, I'm not even sure. So now, if you try sort on this, and given the fact that the algorithm is not exactly deterministic, there are other issues. Are you going to be able to take another bio and next day do the same thing and sort in exactly the same way and see exactly the same result? Maybe if it's a linear transformation, but what if you have UMAP and you sort in the UMAP space, right? You gate it in the UMAP space. This may be very problematic. So I think that a part of time and computational power and data issues, the other big problem is we have to be more serious into discussing the underlying representation of the data. What is actually showing up when we do manifold learning models?
All right.
All right.
Quick coffee break because we're just a hair bit behind. That's not on any of our speakers. Let's try to make that in 15 minutes. So what do you say? 2:10? I'm sorry. 3:10. 3:10. Yeah.
That's it.
Cool.
Thank you. Right. Well, it definitely opens. The question is like, how much? We've had luck. Yeah, where should I put it on the desktop? Let's see if we can. I just put it on the desktop. Oh yeah, I don't. Thank you.
I said I would if I had to.
Oh, stop it here. No. Right.
Right. So we just do
Yes, you could just, if you go to
This?
Zoom.
and share. Can I make this like a presenter view?
You
No?
cannot.
Oh,
It's
why?
only mirroring the screen, or actually it registers the screen
Oh,
as the same.
are you sure?
Yeah.
Oh, I didn't know that. All right.
But I'll share it on Zoom.
We streaming and everything already, or?
We're just about to.
Right. Give a little read. I'm just gonna give you a quick introduction. All right, everyone, we are in the home stretch. Last part of the session today, and it is my pleasure to introduce Anna Belkina. Anna was an currently assistant professor at pathology and laboratory medicine, and the associate director of flow cytometry core facility at Boston University School of Medicine. She received her MD from Russia State Medical University in Moscow and her PhD from Boston University School of Medicine, investigating the epigenetic regulation of inflammatory responses driven by bromodomain proteins. Anna's research is focused on the intersection of immunology and computational biology. Her current research efforts include investigating the immune landscape of chronic inflammatory diseases and developing computational techniques to assess high-parameter single-cell cytometry data. Anna is an active member of ISAC and has been named 2015 to 2019 ISAC SRL Emerging Leader, and I have a privilege of also serving with her on the council, which is absolutely wonderful. So without further ado.
Thank you, thank you. It was probably my fault. I didn't update my bio, so I'm actually running the flow core now. So I've been here in 2018, so some time have passed. Yeah. So anyway, so I thought that probably everybody's eager for lots of math and discussing clustering algorithms and comparison of their machinery, right? Yes, please. No, we're not going to do that. So, sorry to disappoint. So I thought that for like the last session and the last day, we're gonna do a little bit more like philosophical discussion of what we do with cytometry data and how we leverage computational tools and what we do when they produce results that we're absolutely not expecting in a bad way. Okay. So, and I called this talk like, are those clusters in the room with us right now? Because a lot of times I hear people asking like, oh, is this cluster real? Like, oh, I see clusters here. It's like, yeah. All right. So just to start, right, we know that we have amazing advances in the multi-parameter cytometry unrolling on the last decade, and lots of new advances were done in the instrumentation and in the reagents, but obviously the data analysis tools are picking up, and now you can actually see that pretty much all major industry partners are also participating in producing platforms that would allow us to do the data analysis tools and not just leave it to the independent development from some mathematicians who are generous enough to share their algorithms with us, right? So we can measure numerous parameters per cell, generate high-dimensional single-cell data sets, but we have challenges with that, right? So we have the ways we deal with data for many, many years, like manual gating, which now people say is inefficient and biased, and it's intrinsic. It was always biased because it was done manually by the researchers who put their perception of the biological concepts into their analysis, right? So also, even if we think that we are absolutely unbiased and we take our data critically, we still usually fail to leverage the full potential of acquired data once the data sets become large enough. We just don't have enough time in the day, and like slavery is prohibited in the United States. You can't have 20 students do manual gating for you and generate data and compare the p-values to find the gates that are actually producing meaningful results, right? So, and as the data sets are increasing in complexity, these problems will only go further and further, right? So, and I like to show this slide that I picked from Josef Spidlin. Hi, Josef. Who was showing like the tendencies in the computational hardware that we can leverage to analyze the data. Like, and obviously there are major advances in that, and some of them were discussed by Bartek earlier today. So we go from like, we can now go through like multiple samples, thousands of samples, millions of cells. We measure many dimensions, many features in these data sets, right? We used to build up our computers and purchase like the best Mac stations to have max RAM and put GPU in that, but now a lot of people just outsource this into the cloud because it's just not sustainable to do this analysis on your own machine in your lab, right? So, but we have ways to expand there. However, there was not much evolution going on over the last 30 years in the human brain department. I mean, although they're like small changes, actually, if you talk about evolutionary biologists, they will tell you that actually technology is changing our human population. We're getting like some new features, but I mean, pretty much we're same as before cytometry was invented, right? So, and if it was easy to grasp certain things in 1985, right, these things are still easy to grasp now, but the new challenges, the multidimensional data, are not that easy to grasp by our brain power. And I think that sometimes one of the challenges for people is to kind of understand their limitation in that sense, because it used to work, right? It used to work when we had a 6 color panels, right? Why stop working now, right? But yeah, and we're not getting chips implemented in our brain, so it's not going to get any better anytime soon. And we need to do something else about it, right? So I made this talk basically mostly inspired by the insights that I got from several computational workshops that I did over the last couple of years, including workshop at Cyto where we were talking about what actually prevents people from starting to use computational tools. And I'm going to talk a little bit about the results of that workshop and how we actually bridge the traditional analysis methods with the high dimensional data analysis, how hard it is, okay? And how actually we align the outputs that we're measuring with the computational tools with like philosophy of handling cytometry data. And last but not least, which is not on this slide, is some recent Purdue discussions. So, which was about like, what's cluster? And I highly recommend, if you haven't read it, like go ahead and read it. It's a very fascinating discussion, and some people in this room actually took part in it. But what I found interesting is that, I mean, I saw a lot of like pet peeves, some of them that I share actually, so spoken up, but nobody actually talked about the fact that our cytometry data, our data sets are like special in a very particular ways. They're constructed in a very particular ways. And they are constructed using the bias of the researcher who designs the experiment, designs the panel, but that at some point the moment you start to apply computational tools, you somehow think that it kind of like clears that bias away, and you can use the tools that were developed for data sets that are way more like natural to apply to this very heavily engineered data, and to understand what's the engineered part of your data, I think is the key to understanding how you can apply computational tools. Like, what exactly are you tweaking there? Like, because, you know, like I look at my phone, for example, and I look at the pictures that I took of myself, and some of them are like, oh, I looked good on that day. But actually, like if I make a selfie, I have like very subconscious way to like do it right, right? So I don't really think about it, but it's actually very carefully constructed to not show some of my features that I don't like, to kind of like hide the fact that I don't like how my hair looked at that day or things like that. But then several days later, I look at this like, yeah, that looked good, right? So we tend to not notice such things, and that actually basically creates an impediment for us to apply unbiased data methods. Okay. All right. So, and a lot of times people are just like very critical about themselves, and they're trying to do that, and they're just saying, well, am I just doing it wrong? Maybe I'm dumb. Maybe I'm not a bad statistician. There is a proper way to do this with flow cytometry data that probably someone knows, like Bartek knows, right? Like, I don't know how to do that, but Bartek knows. He can actually use this algorithm to produce exactly the readouts from my data that would be correct and that I would like, right? So, but the answer is that no, it's not that like you're doing it wrong, right? It's just like very likely there's no right way to do it, and you're just hitting the wall with that. Okay. So when we were asking people what are the obstacles to success in using computational tools, like we were interested in like all different aspects of like what prevents people from getting started with computational tools, what's the problem of selecting a right tool, accessing it, how to use it, do you like the output, do you have resources in your institution to actually run these algorithms or to get education to how to do that. So all these different aspects. So we'll probably write some kind of report on that and submit it to Cytometry. But I wanted to highlight a couple of things of like, I mean, we had a bunch of readouts of this. We had a big survey, multiple people answered. So so I wanted to highlight some common themes in what we saw, right? So I'm not talking about like how you get started, right? So that's pretty much expected, what you expect to see. But when people talk about how they select the proper tool to do computational analysis, sometimes they are just like, well, there's so many options. I can't pick one because there's a right one. Among them, there is a correct one, but I don't know which one is correct, right? And I'm not sure if it fits my question, but I'm sure there is a tool that actually fits my question. I just don't know which one it is, right? And then also like, well, I know that my data quality can be maybe questionable, but I'm not sure if the algorithm will actually tolerate that, right? So I didn't realize that I had an animation there. So just watch the green part. So the green is basically people who agree with the statement. So more than half of the people say that they are not sure if the tools that they have provide the appropriate analysis for their question. And that's actually a very good point. Like it's correct to be not sure about it, okay? And people who try to start doing the computational analysis, they usually have several options. They can either build their own toolkit, they can use the existing toolkits that the software packages provide, or they can outsource it to someone who knows how to do that. And they can say, well, Anna, would you like a collaboration? What if I dump this, multi-gigabyte data set on you and you produce this beautiful OptC map of the data, and everybody would benefit from that? So, I mean, that's an option, definitely, that you can do that. But I think that understanding the analysis workflow principles is way more important than choosing the specific toolbox and deciding which algorithm to use, because the difficulty is that you need to learn what to do and what not to do and how to do it at the same time. So that's the biggest obstacle that people see, right? And when they get to like how the algorithm performs in their hands, A lot of times people say, well, the results are not consistent with my expectations. I'm not getting the results that I want. And I know that there are a bunch of parameters there. I can tweak different knobs, and I'm not sure which ones are correct, right? So we don't know how this works, but what do we do, right? So, and then finally, when they look at what the result they're getting, they're like, I don't know how to interpret this result. The result really doesn't agree with the whole bunch of other research that I did. It doesn't fit. I had like a 7-color panel, and I expect 10 populations there, but I got 59 clusters. What do I do with this, right? And the power of this analysis, I tried the statistical tools that are recommended. It's just not powerful enough, right? So many, many, many obstacles. So let's go back and talk about what we take for granted in cytometry data, okay? So first of all, when we design our cytometry experiments, right? Even if we go to like 50 colors or 50 metals with mass cytometry, it's actually like when people talk about high dimensional analysis outside of cytometry world, they don't mean 50 parameters, okay? So that's not. So we cherry pick our markers because we cannot afford measuring noise, measuring something that's not meaningful for our readouts. And even if we build a 50-color panel, we're still cherry picking parameters because there's way more CD molecules than 50, right? So we're still selecting things that we believe would be helpful, okay? That have proven relevance for research goals, that have minimal redundancy. And if you're measuring, like if you look at, for example, transcriptomics data, there's high redundancy in that data, and that's actually good. So the data supports itself, right? But we have very little redundancy usually in our data. And each of the like mini assays that we run when we combine the flow cytometry panel reagents together is optimized in limited context of known cell populations. Even if we're going into the like exploratory mode, we're using markers that like, okay, we know that CD5 is a marker for T cells, right? Do we know what it actually does on other cell types? Well, it will be a big surprise for you to find that actually it's important for B cells. It's an even bigger surprise for a B cell researcher who wanted CD5 to find B1, B2 cells, to find that it actually blasts out all their T cells because it's expressed on pretty much every T cell. Okay, so sometimes when we design our panel, there's all this rules of like primary, secondary, tertiary antigens, you know that, right? So something that's primary antigen for one cell type is actually like totally tertiary antigen for another cell type, which is basically means that the readout is noisy, uninterpretable. We don't know if it's important or not, but it can actually spoil your data pretty badly, right? And why do you not know about it? Because there was no single person in the world that would use a purely B cell marker in a T cell panel, you know, until 3 years ago when people started to heavily use spectral cytometry. People just didn't do that. Like if there is a primary marker for T cells, it will be in T cell panel. Nobody will put it on B cell panel. People cannot afford it. You can't afford to spend a PMT on that, right? So when we plan which markers we measure, we already have expectation about the distributions of the expression across the range of detection, right? So we know that something is like very bright, something is very dim. We assign it to different colors, right? But our clustering algorithm doesn't know all that, right? So when it gets to the clustering algorithm, it assesses the signal that you're getting for every cell in the data set, and what is like one man's treasure and a nice marker to delineate something becomes completely noisy input into the subsetting of another cell type that you're running inside the same data set and using the same algorithm, okay? And then also we have huge knowledge of biological, like immunological knowledge, right? We kind of like, we have a range of like, this is an important marker, this is less important marker, this is, oh, this is prominent, this is not prominent. This has been published like in 1980s, maybe even. Again, clustering algorithm doesn't know all that. And if you have super important marker, which is actually, in fact, like the detection is not that great, it will treat it in an unbiased manner, which is basically, oh, there's some noise there, right? I don't believe it's like, it's a very important PD1. Well, yeah, I know, but it's actually like a smear that I cannot subset from anything, because the variance that you're having in the positive and negative is overlapping so much that you can't really like see whether there's, it could be actually a primary antigen in terms of like it's on off for certain cells, but you're actually getting a smear, right? And when someone takes the big data set and has starting the manual analysis of it, meaning like you're subsetting the data, right? So you're almost clustering, right? So you're subsetting data into certain groupings. You conveniently ignore features and measurements that are poor quality, right? So if something is, I mean, I kind of reiterating what I said before, if something is important for one lineage, but is noisy and not helpful for another lineage, you can just ignore that fact and not notice it until you feed this data into the clustering algorithm. So if the biological knowledge that we possess makes it irrelevant, that's totally okay to ignore it. But again, you cannot inform your clustering algorithm with that. Why can't I transition? Here we go, right? So things that make data hard or easy to cluster, some of that, like what we think the cytometry data are actually like, they should be easy to cluster, right? We can have lots of variation in the data, that's nice, right? Some of the readouts are pretty low noise, that's great. We think we're doing high dimensional analysis, that's cute, okay. And we can generate actually lots of data points pretty easily, right? So if your data have little variation, what we call just like, oh, well, every marker I'm measuring is smeary. If there's high noise, if it's low dimensional and sample size is low, it would be really hard to cluster. And sometimes you start with a panel that had lots of data points, lots of variation and things like that, but actually you gate it first to the population you're interested in. So I take my CD3, CD4, CD8, CD25, CD127. I find my Tregs, I gate on them, and then I say, dear clustering algorithm, can you cluster based on the transcription factors that these Tregs make? And produce me a beautiful map of how these cells have different lineages. And the results will not be what I expect from my biological knowledge of, oh, there's actually very distinct populations there. Because the data that I feed, I don't feed my nice markers in there that have a bimodal distribution. I feed some kind of like noisy smears in that. And then you don't get the result. And then you try, oh, well, I'll do dimensionality reduction to show this, and you get one big blob. And that's when you say, well, computational tools just don't. So just to summarize, right, what prevents our clustering results from meeting our expectations? When we have complex marker dynamics, so varied expression patterns across populations. Some of them, for some populations, they would be meaningful, but for some, they would just produce errors, right? Some markers that we consider super essential based on our immunological knowledge can actually be not really flowable, right? So may not translate into clear clustering distinctions. And if you force the clustering algorithm to show you the distinction, usually the result will absolutely not be what you expect it to be, right? And it's hard to find a one-size-fits-all. So there's no middle ground that would allow you to filter the noise and still maintain biological patterns, and all done in one algorithm. So at this point, I'm like super pessimistic, right? Everything is bad, nothing works. Well, no, things actually work, but I just wanted to try to explain why when we start approaching clustering analysis, and we see people successfully showing their beautiful clusters, why your result may not be same and how much work actually went into making this work, okay? So the clustering algorithm will not do things that a human would, right? It will not disregard features for some cells and use them for others. What you can do and you can control is that not everything that you measure in your data set should be fed into your clustering algorithm. Absolutely. You need to be critical. Like, even if you're like, okay, I'm already only 20 colors, I want to use every scrape of data that I have. No, you can be critical on that level. But sometimes, unfortunately, yeah, you have the same marker that you know that if you remove it, it will make one population cleaner and another population not distinguishable. So that's it. It means that you hit the wall of not having enough redundancy in your assay. If you could have another marker that would distinguish these populations, that would be good. Think about building it into your pan, okay? You cannot infuse biological significance into the interpretation of your clustering. So your clustering algorithm will not make any interpretations for you, right? While the person who is gating has usually the idea of what they're gating, right? They take the gating strategy from the literature, and they know like this tree branch is important and this tree branch is gibberish. But the clustering algorithm will not do that, right? And finally, if you have imperfections in your data, a lot of times like manual gating will adjust to that. You're analyzing the data set, you're going like sample to sample, and you're shifting the gaze just a little bit because, well, it always makes sense. Like, you know that this is CD4s and CD8s, and even if they shift quite a bit, it's like biological knowledge guides you where to put your gate, right? The clustering algorithm will say, well, there's one sample with CD3 median at 10 to the 3 and another with CD3 medium at 10 to the 4. These are obviously two different sets of T cells, right? So I'll put them in two different clusters. I mean, if you're using the metric that would accommodate that, obviously. But maybe it was just like a batch effect and you just didn't add enough antibody in one of your stainings, right? And the manual gator will actually accommodate for that easily without thinking, right? So the algorithm will not ignore the data. So if you're trying to mimic the thinking of the human analyst, this is usually where the problem stands, okay? But what the clustering algorithm will do is what's on the can. And like pretty much like there's so many of them. I'm not going into like showing you zillion examples of different algorithms, but this is where I would say, go back to the Purdue list and read that thread where people were discussing like what's a cluster, right? So the clustering algorithm will assess how similar cells are in multidimensional space and then assign them into the clusters based on certain measurement of their proximity in multidimensional space. And every clustering algorithm will do it in its own way using multiple metrics that exist. So you can, if you understand how this works, you can pick the algorithm based on the metrics that it's using, pick it for the task. But yeah, that's what it will do, right? We're doing well with time? Because we started later. I just want to make sure that I'm not. Okay. Okay, so what is actually messing up our clustering and sometimes gating, right? If you have pre-processing issues that disrupt clustering, if you have inconsistent flow in your data, so variability in signal during the measurement, that's a problem. If you have compensation, unmixing artifacts, that will be a problem. If you have noise, if you're including debris, dead cells, doublets into your data set, like your clustering algorithm may actually pick it up and subset those data points into clusters, right? Or maybe not. We don't know. But if you want to limit this, you know, in your clustering algorithm, you can actually clean it up beforehand. And then also, usually most of the implementations that are granted to you by if you're not writing your own code, if you're not taking someone else's code and read it, right? So usually you can presume that you're doing in your analysis software, you're doing certain transformations of the data, and those would be carried over into the clustering algorithm performance. So if you scaled your data in an awkward manner, non-traditional way, or if you messed up the transformation values, like you can get absolutely inadequate results, okay? And if you have one batch of samples or like one sample, that doesn't apply, but if you have multiple samples, batch effects would definitely compromise consistency of your clustering. So if you put all your samples from multiple batches together, you may actually find that your clustering algorithm picks up batch distinctions as a signal, right? And that's definitely not what you want to happen. A couple of examples. The first flow slide in the whole talk, right? So this is an example of like unmixing artifacts in the data generated by Chris Hall. You can imagine that this population that's actually completely non-existent will actually appear in multiple clustering data set output, clustering algorithm data output as a cluster or something like that. So if you have noise that you didn't remove that originates from unmixing artifacts or compensation artifacts, it will show up in your data. This is another example that I showed in the talk last year for like crimes against cytometry. So this is publicly available data at Flow repository. So same person, same subject, 2 days apart, two blood draws. And on the second day, there's a new population in their PBMCs. And it's actually not a new population. It's actually a complete artifact of inconsistent flow. So things like that, if you don't control for this, you will have artifacts in your data. This is an example from Jeff Cracker. Thank you. Actually, he had this, like, what you see is what UMAP sees, but not only UMAP. UMAP is not a clustering algorithm, but this is an example of UMAP visualization. So clustering algorithm will also likely consider that there's certain distance between these two groupings of dots. So maybe it's actually two different clusters, depending on how you choose your algorithm. But you don't want to create this kind of puzzles for your algorithm. Okay, so how do you choose finally what you do? Which algorithm is best, right? That's like a big question. Like, what algorithm should I use? Find the algorithm that you can use, so that is accessible for you, and you can actually run without like getting a grant to build a cluster of computers, without enlisting 3 biostatisticians in your team. So something that you can actually play with is you need to get it right. Make sure that your data are scalable. So you can actually put them into the, you can either downsample your data without putting at risk your rare populations, or you take the algorithm that actually can take your full data set. A lot of times, if the algorithms are taken from some outside of the cytometry world, it could be that they're just not built to support that many data points. So don't be surprised. Data set, like if you're taking an algorithm from, I don't know, single cell transcriptomics, you like most of the times you may have like, there are some data sets that are millions of cells, they appear like later, but generally a lot of algorithms are built for like 10,000 data points. That's what was benchmarked. So if you're taking the algorithm that you're using the algorithm that's built into one of the cytometry platforms for analysis platforms, likely they already thought about this. So you can expect that it will work, right? Super important, match the algorithm that you use to your research question. And I will talk about this in a second, but after you figure that out, then you can start thinking about the performance of the algorithm, what kind of trade-offs different parameterization will bring, and things like that. And this is where you may want to seek some help from the experts, right? But you get to that point only after you consider the previous things, and especially the research question, right? There are several publications. This is the one that comes up most often about like comparison of different clustering methods. You need to understand how clustering algorithms work to read this paper, honestly, because it suggests use like multiple metrics, how people assess clustering algorithms, but doesn't give you an answer, clear answer, like what algorithm is best, right? But if you're interested in looking at, oh, so how do people actually assess whether the algorithm is doing what it's supposed to do, whether it's working for a certain type of data, read that paper. That's a really good paper that will kind of give you a framework for thinking about this. Okay, so how do you match the algorithm to the research question? You usually with cytometry, you do one of the few things. You want to identify novel cell phenotypes. You want to characterize known immune populations. Oops. And then there are like a couple of other applications. But if you're looking into identifying novel cell phenotypes, you want to make sure that the measurements that you're taking are very clear-cut biologically, okay? So if, again, like you're looking at the system of this primary antigens in your panel design, and there's the measurement is clean enough for the manual gating, for example, to be like on, I mean, Ryan Brickman has ideas about like what's easy to gate, what's not. But let's take like our mere human take on that, right? So if you're trying to identify a novel phenotype and it's just like a smearish smear of your marker, of super important marker that you know that has biological significance, well, the clustering algorithm will not share your joy on that. So you want to have something more clear cut. And clustering algorithm will not, if you make the algorithm to cluster the cells that you consider important into a separate grouping, doesn't mean that it proved that you actually found a new population. You tricked it into, maybe you were lucky in doing that, but it's not proving by itself that it's a real biological phenomenon. If you're characterizing known immune populations, and you're like, okay, this is like the example that I'm going to show later. I'm going to look at the inhibitor receptors on multiple varieties of T cells. Don't feed the inhibitor receptor readout into your clustering algorithm. Feed your lineage markers, sublineage markers, characterize your tree so that you have like the biologically proven populations. The clustering algorithm finds them, great. So, but you know that they're real because there's a whole body of knowledge about their existence. And then see what's the expression of your inhibitor receptors across these populations that were also identified by clustering algorithm. Luckily, maybe just like that's good, right? But anyway, you could do this with manual gating, yes. You want to do it with clustering, fine. But it's not proving anything, right? So this characterization, you don't want to put it as an input of the clustering and as an output, because you can't do both. You can do either one or another, okay? And then finally, people want to use this to understand what kind of heterogeneity arises in response to certain experimental intervention, treatment, or things like that, right? So I have new population rising when I treat my cells with X and Y, or this population disappears, or this population upregulates something. So you have to thread very carefully when you get into differential analysis. I generally think that pretty much every time you want to match differential analysis, so finding differences in cohorts with clustering, this is the time you need to call your biostatistician. And like, I mean, I've seen everything from like just throwing p-values and t-tests on every cluster comparison pairwise, to doing some pretty sophisticated things and using you know, especially if it's like packaged nicely so that you can actually, with one press of the button, you can, oh, I will run this algorithm and it will prove me what I want to see. If you don't understand how this works, ask your biostatistician. By the time you put this in publication, there's a good chance that the person who knows statistics will read this and criticize you to shreds, okay? So I am not a biostatistician. I will talk to biostatistician about this, and I'm not teaching you how to do this right. I'm just saying that, you know, read very carefully, okay? So make sure you have a statistical analysis plan. It's not trivial. Most people do it wrong. If you look at the publications, what people have used, most people did it, okay? So I see Bartek finally looking at me like, yes. So most people do it wrong, myself included. Actually, we have a paper came out that I'm absolutely not happy how the statistics were turned out, and I couldn't argue with some collaborators who wanted to do it certainly. But work with biostatistician if you're not a biostatistician, okay? And then finally, is that okay? Time-wise, good? Perfect. How you present your clustering. Think about your visualization. So visualizations are really important, and that's what we will show in your slides, actually. So that's how people will judge if you're a good scientist or not, based on like stupid visualization of your data. So it allows you to comprehend this huge amounts of data that we generate. You can change the perception of your data based on how you present them, right? You can actually troubleshoot your data using visualization. So that's all super, super important. Think about how you present your clustering data. And I wanted to show an example, and I also will show like a pet peeve about visualizations that a lot of people share. So we have an experiment where we are looking at the gamma delta subset in PBMCs in a cohort of subjects that either have or either live with HIV or not, and also are of different ages. So we designed a 22 color spectral panel. That was like, don't judge me, it was back when we had only 4 lasers on the floor. So only 22 colors. And we basically wanted to gate on gamma delta T cells and then pull them out of the data set and then see what kind of subsets are in there, because they're absolutely understudied. So, and we find them super cool. So this is the markers that we measure. So we have the markers that we think would be expressed on gamma delta T cells. So we have gamma delta DCR chains, and then we have some other receptors. Then we wanted to look at inhibitor receptor expression on, sorry, CD13 should go here. But anyway, we had inhibitor receptor expression that we wanted to assess, because that's what actually my lab does, and we've been publishing on that. And then some of the things that we used upstate, like in the gating process, excluding CD14 positive cells, excluding CD19 positive cells. So they obviously were not used for dimensional reduction. So now this is an OptiSNE map of 96 samples out of like, I think 12 batches. It's crazy. Can you see clusters in this data presentation? Let me make it easier. So this is the heat maps of expression of, so we have V delta 1 and V delta 2. They're like mutually exclusive, V gamma 9 chain, and then some other markers, and then inhibitor receptors that I said didn't go into the dimensional reduction. Can you now see the clusters in this presentation? How many clusters do you think? 2. Okay, many, right? So there are no clusters presented in this. I will not talk about this cluster clearly represent gamma delta T cells VD1 expressing, or this is the cluster for VD2 cells. There's no clusters here. Please don't say there are clusters here, okay? So, I mean, I can use my neural network and mimic the behavior of a clustering algorithm and think, well, these cells are actually quite distant from those, so maybe they form a cluster. And that's exactly what some clustering algorithms do, right? I mean, I'm looking at 2-dimensional representation, but they would look at multidimensional. I cannot imagine 22-dimensional space with my poor neural network. But let's say I'm just looking at this, right? But that's just me imitating clustering. This is not clustering. Now, I used Phenograph, a clustering algorithm, to clusterize this data. And Phenograph found 39 clusters in this data. So do we think there are 39 subsets of gamma delta T cells in the compendium of this 96 subject blood? No. We don't think there are like 39 different biologically relevant, or like some of them are similar populations. We think that Phenograph found 39 clusters. I can visualize them by overlaying them with color. And if I put it next to the heat maps that I just showed, I can say, well, some of these clusters found by Phenograph comprise the biological population, that's VD1s, and some comprise biological population, that's VD2s, okay? Now, I can visualize this with heat map, and this is how usually people visualize data from clustering algorithms. So I will put heat map where this is the numbers of all clusters, identifiers, and this is the markers that we want to visualize. And some of them were actually used for clustering, and inhibitor receptors were not, because as I said, I didn't want to include them. I want to actually characterize the clusters that I identify, how they express different inhibitor receptors. So I can say that, well, these are all VD1 positive clusters, right? Do we think that there's like half of like 20 VD1 positive subsets there? No. I can look for certain patterns, and because I know each of the markers that I cherry-picked for their biological meaning, I can imply that there can be certain meanings in these clusters, and some can be completely meaningless. Okay, and I'm totally prepared for that. So next thing I want to do with that, I'm looking at this and I'm like, okay, I'm interested in certain subsets. So I will design a different experiment that will actually allow me to assess the biological validity of this cluster. So if I stop here, that would be really bad. And this is where a lot of people stop. Okay, so this is a quote from Dave Noble. Some clusters will mostly by luck be biologically meaningful, and others will not. So if you're asking, well, will this clustering algorithm find my new phenotype? Well, you don't have a new phenotype, okay? You need to prove that it exists. Sort the cells. I want to sort this panel on a spectral sorter. Sort the cells and do some kind of orthogonal assay. Make them produce cytokines. Sequence them. Do something. Right? But don't really use the clustering algorithm as your biological readout. Another example, just how many clusters is here, right? So this is another, this is my last slide. So this is another example where I have, I applied also Phenograph clustering to a subset of fetal human liver cells, hematopoietic stem cells from fetal human liver. And here it's even more difficult because in the gamma delta data set, I mean, biologically, I know it's pretty flat. So there's like a lot of subsets that are not really forming a tree, right? So here, it's more like a continuum of developing hematopoietic stem cells that are slowly becoming differentiated hematopoietic cells. So the clustering that I do is just slicing through the trajectory of continuum. And all I can deduce from this is that I'm looking at behavior of certain markers, and I can think, well, these markers tend to be in the biological population that I expected by designing this panel. So I need to use more tools and more analytical approaches to actually assess this continuum. But the clustering itself here is actually kind of against the biological logic of this data set. And it's fine, as long as I know what I'm doing. And with that, I just want to summarize what I just said. If the clustering algorithm is not working for you, it's probably not you, and it's not the algorithm, it's the combination of the special design of your data, the deficiencies of the algorithm, and maybe deficiencies of the operator. But when you try to approach it from the side of deficiencies of the operator, and maybe the deficiencies of the algorithm, that will be a dead end in many cases because you really started from the wrong step, okay? And think about it wisely before you start to push your clustering interpretation into biological significances, because a lot of time you're just looking at dirt and you're finding patterns in the dirt. And there's a word for that. Okay, that's it. Yes. Sorry, pairing of what? Height, width, and area? Okay, so the thoughts of using height, width, and area of the electronic pulse for clustering purposes? Well, I'm an immunologist and I study lymphocytes, so I'm not an expert in that because it all measures the same for me. I'm pretty sure that you can deduce something extra from those features, but I'm pretty sure that would be like going to the standards of difficult to cluster concepts on my list, right? So you will probably see some kind of difference, but it's not, you don't have the distribution there that is easily revealed by some clustering algorithm. Yes. I know where you're going, yes.
But then when you squeeze it out, but when you squeeze it out and do rank reduction, right? Suddenly it's also 20, like in our case, but we are not so, uptight about it, right? Because we had 20 from the beginning. And this is related to what Daniel was saying, that you could take all the physical parameters as well, and you would increase the dimensionality, right? But
the hidden dimensionality remains kind of same.
It could be the same, right? It could be that nothing is there, but given what the type of cells he's working on, right? Maybe the shape, maybe the other things would actually contribute, and then we increase the dimensionality and we increase information. So basically my comment is that we are not such low dimensionality as other omics people would like us to think.
I certainly agree. And I like to tease people who bring me single cell transcriptomics data. And they're like, well, we measured 3,000 genes. And I said, well, but your UMAP that you're showing me is from 20 PCA components. And this is the real dimensionality of your data. Right. So, and that's why it actually looks strikingly similar to visualizations of flow cytometry data sometimes. And I mean, after the OPSNI paper was published, it's been used in so many different fields. So every time I see like someone citing the paper, I'm sometimes curious to see how they use it for completely not biological data, right? And we kind of like, we are used to certain looks, right? So some things like just look completely wrong. But because we have never seen how astronomical data are basically dimensionality reduced or clustered. We just see our biological data. But yeah, I totally agree that, I mean, the hidden dimensionality of my 22 color panel is probably like, yeah, maybe 10, right? And when you have a truly 50 marker measurement, it actually may not fit nicely into, for example, Optisne space. And if you try to UMAP, it may be better to UMAP into like 3D and not into 2D because it doesn't fit anymore. So
Suddenly, UMAP is not enough to represent your data.
well, we can still think in 3D, you know? So we can think in 4D, but 3D is still imaginable. So we have some leeway. But yeah. Is Bill's presentation here? Oh, you'll manage. Thank you.
Hello, everybody. Our, I believe, concluding speaker. This is going to be Bill Telford. He got his PhD in microbiology from Michigan State, where he developed some of the other Michigan States. Okay, you can clap. Okay. And get them next year. Where he worked on developing some of the techniques of apoptosis assays. He did his postdoc at the University of Michigan Medical School. He's since become a staff scientist at the National Cancer Institute and the NIH in 1999. Now he's a senior scientific associate and director of the flow cytometry core laboratory at the NCI Laboratory of Pathology. Currently, his research interests include advanced instrument and assay design aimed at solving unusual problems in flow cytometry.
Thank you, Jake. Sorry, I'm losing my voice. I always do at the end of the. Thank you for having me. Thank you to the organizing committee and Joel and Rui and Meredith for inviting me. This is not a computational cytometry talk. It's entirely my fault that it's not because don't cheer. That's not a good reason to cheer. I couldn't come the day they wanted me to come, so they put me on the third day, which was very gracious of them. So I run a core lab in the NCI, and what I want to talk, and there is no four color data today, but I do want to talk a little bit about how we might be able to push beyond the 40 to 50 color limit in flow cytometry, both conventional and in spectral. So this is going to be really a technology talk about lasers, which is something we do quite a bit with. And as everyone in this room knows, we've sort of settled on these 5 wavelengths: the blue, green, the red, the violet, either a green or a yellow, and an ultraviolet. We've got lots of dyes right now to pick from. So we can do 30 colors, theoretically, by conventional flow. We can certainly, I mean, you've seen plenty of 20 and 30 plus color data at this meeting. We can go over 40. And if you're going to Edinburgh, you'll see data up to 50. But what if we want to go beyond this? What additional laser wavelengths are we going to need? Because I think that's really going to be the path that we're going to have to take. We're going to have to add additional laser wavelengths. Now, I've been doing flow for a long time, and although UV lasers have always been part of flow cytometry, the development of the BD Horizon BUV dyes really made them extremely important. Now we can't do high dimensional flow without them. It's a critical wavelength for cytometry. We've got the BioRad, the Starbright dyes. We have other manufacturers who are starting to provide these. So not only can we use the UV to add to our high dimensional panels, we can do it in a pretty dramatic way. We can add 5, 6, 7 colors or even more. And if we're doing spectral, we can even combine these guys together and really, really take full advantage of the UV. However, I think it's important to remember it's not easy to generate UV. And even today, the solid state 355s that we use are not easy to build. It's still a pretty tough technology to generate, although we have certainly more powerful ones now. We have small ones like the coherent, the Obis 355. Cobalt in Sweden makes a little 355. For years, I tried to get people excited about the 375, which is a much cheaper diode source. I completely failed at that because they're in a few machines, not very many. Yes, the Beckman-Coulter Cytoflex, thank you. You are the only commercial instrument I know that has one. Yeah, that didn't work real well, unfortunately. But anyway, we certainly have the technology for this. And now we have some alternatives in the what we would call the classical UV space. The Coherent has built a 349 and a 360. These are much easier lasers. I wouldn't say much, but they're easier to build and they're cheaper. So we can now get UV without as much expense and without as much manufacturing difficulty. And we've tested them running the BUV dyes. These are staining indices here. And although you might not be aware of it because the manufacturer knows we love our legacy wavelengths and we don't want to change, you're going to start seeing these creeping into instruments because they're quite reliable and they operate pretty much the same. So we need an additional wavelength. And what I propose, and I've been proposing this for 6 or 7 years now, is that we could add, because these are really the standard 5. Some people have yellows, some have blues, but they really don't add high dimensionality. They only add specialized fluorochromes. Can we add a 6th one? And I've proposed the fact that really the deep ultraviolet is the only place for us to go in this for a number of reasons. And I'll go through some of the reasons and I'll show you some of the technology. A lot of this has been published, a lot I've presented before. It's DUV laser technology, I'll tell you up front, it's hard to do. These are not easy lasers to build, but the technology is starting to mature to the point where we can get it into flow cytometers. So really, if we're going to go above 40 colors by spectral, we have to add additional laser wavelengths. And although some of us have near IR lasers on our machines, we're not going to get a lot more colors out of that, maybe 1 or 2. Fluorescence starts to fail as a phenomena as you move up into the near IR and the IR. However, for deep UV, fluorescence still very much functions, often when we don't want it to, as in cellular autofluorescence. But there is no physicochemical reason why fluorochromes that excite below 355, and I'm defining deep UV here from about 215 up to about 320, there's no reason these cannot be built. They don't really exist yet, but they certainly could be built. And as everyone in this room knows, one of the wonderful things about spectral cytometry is even if you don't have deep UV fluorochromes, you might still benefit from having a deep UV laser on your instrument. You can have a UV laser on your Aurora or on your ID 7000 and never run a UV excited fluorochrome, and yet that laser is still going to excite all your other fluorochromes and provide additional data, additional information for your spectral unmixing. Now, there are a lot of these out there yet, but I will give you my list, which I update periodically. Many of you, I think, have heard of the 320. This is a laser that is starting to appear in cytometers, and we'll talk a little bit about that. It's a tough one to build, and we've gone through a number of manufacturers. You can dip down to about 303, and you can also get into the 200s, and we're actually getting quite low, down as low as 213. Lasers are on this list because they are, at least in theory, applicable for cytometry. They're not pulsed, they are CW, they're not 10 watts, which is typically where deep UV lasers are. 266 nanometer lasers are used a lot in manufacturing and quality control for superconductors. But these theoretically can be introduced into cytometers. Now, we have an instrument set up for this. This is my 22-year-old LSR2, and it still works. But we've modified this instrument to be compatible with deep UV sources, because as you move into the UV, the materials you need, the mirrors, the optics, the lenses, you need to start changing materials. So this instrument is equipped with aluminum mirrors, with fused silica optics. But really the modifications you need down to about 250 are not at all onerous. They're not very expensive. So it's not that difficult to change out the instrument to accommodate sources below 355 or 355. Once you get below 255, you have to make some additional changes. The coatings have to come off the mirrors. You need calcium fluoride lenses, but again, nothing crazy. It's really not that difficult to integrate deep UV lasers into cytometers. I will mention that once you get down to about 220 or 230, 220, the cuvette starts to be a problem. The fused silica doesn't transmit that anymore. So in our systems, we have to move to a stream and air. But again, these are solvable problems. We also rely heavily for evaluating these lasers on quantum dots. Remember quantum dots? They've seen a minor resurgence in interest just because we're doing spectral now, but we really don't use them much as fluorochromes. However, the great thing about quantum dots is that they excite extremely well in the ultraviolet and deep ultraviolet. They don't crap out in fluorescence as you move into the deep UV. So for us, they make a very good internal positive control to make sure our system is working, that the laser is properly integrated into the cytometer. And then we can go ahead and evaluate more conventional organic dyes. So this is the first thing we do, is check with Q dots to make sure that the laser is working. Now, I've talked about this laser in other presentations. This data is published about 7-8 years ago. Lazos, which is a venerable German laser manufacturer, produced a 320 nanometer laser. So below the 355, we're moving into the deep UV range. For those of you old enough who have been in cytometry long enough to remember HECAD lasers, this was meant to be a HECAD replacement, which emitted at 325. HECAD lasers were big, they were noisy, they didn't live very long. They did appear on cytometers, but they never got used a great deal. But this was meant to be a replacement for that. One nice thing about this laser is it's tiny. It's in the coherent cube format, so very easy to integrate cytometers. And if you analyze Q dots, you will find that the 355 and the 320 are both equally efficient at exciting these fluorophores. So we know that the laser is probably working pretty well. Now, if you go into the Spectral software, fluorescent probe software, and try to estimate how well the 320 will excite, for example, the brilliant ultraviolet dyes, it doesn't predict that it's going to excite them very well. However, I have a feeling that the data for this really isn't in the software. This is an extrapolation because really nobody was bothering to check down that low. And when we look at the brilliant ultraviolet dyes, and again, these are just the staining indices, you can see that the 320 excites them very well, about as well as the 355. This is could be a good or a bad thing. It's a good thing if you're trying to replace your 355 with a smaller laser. However, if you want to add 320 to a 355 in a multi-laser system, it's probably a bad thing because you're going to find it very difficult in terms of cross-beam compensation and spreading to have the laser function that well. Because what we really need, one of the things we need is if we're going to be adding in additional laser wavelengths, the fluorochromes have to be compatible. We want our conventional UV to excite our brilliant ultraviolet or whatever dyes we're using well. And if we design deep ultraviolet dyes, we want it to excite not that well. We want the inverse with a deep UV laser. Otherwise, the amount of compensation, the amount of spillover, and the amount of spread is going to be unacceptable, and we're going to have great difficulties. Part of this is choosing the right wavelength. It's also partly influenced by dye design, and we'll talk about that as well. Because now when manufacturers are building new dyes, they're not just thinking about the dominant wavelength, they're also thinking about the incidental excitation by other wavelengths as well, which is something we have to think about all the time, but we really have to think about it in Spectral. I've never been wild about the 320 because it's awfully close to 355, but I've always taught in terms of conventional cytometry. This assumption may be turned on its head by Spectral. because remember that all of our lasers in Spectral excite all of our dyes. And it turns out that 320 may excite some of our non-UV dyes in unexpected ways. So this is something we'll discuss as well. Now, deep UV dye development really has not occurred simply because we don't have reliable data sources yet. But BD took a stab at it some years ago, and I have permission from BD to show this data. These are actually a series of deep ultraviolet brilliant style core dyes that BD developed in anticipation of having a 300 nanometer laser source. These dyes are not commercially available. They may never be. If a manufacturer does ultimately decide to make deep UV dyes, it will not be these guys. Because the process by which these are developed is quite interesting. They produce many, many potential cores, and they test them for tandem generation. Some tandems might have some cores, some might have others. We'll never find out. We don't need to know. But a lot of work goes into developing these things. So these excite around 300 and they emit around 360, 370, about where BUV 395 emits. And if you look at the 320 does a fairly decent job of exciting these dyes, about as well as the standard BUV 395. What's really amazing about this technology is the fact that the 355 doesn't excite them at all. There's good spectral isolation between these two laser wavelengths. So once these cores are generated, they go ahead and develop tandems. And again, these are not commercially available. Don't ask for them. Anything that ultimately appears will probably be completely different from this. But you can develop tandems to these dyes just as you can to the brilliant ultraviolet. And again, they function quite well. And the 320 does a much better job of exciting them than the 355. So again, good spectral separation between the two of them. In terms of intensity, they're pretty similar to the brilliant ultraviolet dyes over here. But keep in mind that these wavelengths both excite the BUV dyes extremely well, too well. We don't really want them to. Now, unfortunately, Lazos is not really building this laser officially anymore. It turned out to be not a very long-lived laser module. They typically died around 3,000 hours. And for those of us in Flow who are doing high-end stuff, we may not be bothered by that, but these companies are very stringent about what they release to the public. I think they will build them for OEM manufacturers. The Sony ID7000 might have a Lazos module in it. They won't tell me. But there are operational lifetime issues. And as a result, they're not building this anymore. I talked to them at the Photonics West meeting just a few months ago. So we need to get another source. And unfortunately, not many manufacturers are building to this. So I've talked about this company at previous meetings. There's a company in the Czech Republic called Kvant, which you may never have heard of. But if you've been to a concert or a club or a sporting event, the motion-controlled laser light generated at that event very likely came from one of their units. They integrate lasers into display systems. They're very, very good at this, and it's a highly lucrative business. But the guy who owns this company is a physicist, and he's always had a dream in the back of his mind that he wants to build scientific lasers again. So they built us a 320. This is the module over here. It's pretty small. It doesn't work that great. It's buried on my instrument. You can see it's buried under cooling fans and Peltier coolers and all sorts of things to maintain the temperature. The beam shape is very strange. But it does a wonderful job of exciting Q dots, which are always kind of our first line of validation to make sure the instrument is working well. But again, it's not going to be a very good long-term solution. Victor Dubek, who's the owner of the company, has no interest in marketing this laser. Nobody wants to buy it, unfortunately. We don't count. However, there are commercial units out there that you can actually go out and buy. Cobalt, which is a venerable Swedish laser manufacturer. They're owned by the larger company. Hubner has a 320 in production. You can actually buy this laser. It works extremely well. There's a UK company called Skylark that has a 320 module that you can also purchase. It's pretty big, but they're working on a smaller unit. And as many of you know, this laser is starting to creep into commercial systems very slowly. Sony will sell you a 320 that it will strap onto your ID7000. Cytec now has they've actually announced a 320 modification if you really want it. But does it really add functionality to the instrument, or is it more just a 355 replacement? And the problem is when you're working with two wavelengths that are so close together, if you try to run the BUV and the DUV dyes together, and here we're looking at two tandems, BUV 563 and DUV 565, the DUV 565 was designed to not be excited by the 355, but the brilliant ultraviolet dye was not designed to not be excited by a deep ultraviolet. Why would they? It was 10 years ago. Nobody cared about deep ultraviolet back then. So they're not going to be very compatible with one another. There's a lot of spreading, a lot of compensation is required. So my opinion on this laser, and it may change with Spectral, but it's kind of on the line between conventional and deep UV. It's a great 355 replacement. I don't think it's going to be a great 355 add-on. However, that may not be true for Spectral, and we're actually working with Cytec to build a 320 nanometer Aurora right now. So I may be wrong about that. And the nice thing about the 320 is you can actually go out and get one. They're actually available, which you really can't say about many other deep UV lasers. The other issue with deep ultraviolet, and it's not a big problem at 320, but it is going to become a bigger problem as we go lower, is the autofluorescence generated, particularly in the lower wavelength range from about 390 up to 450. It's substantial. And while it's not going to affect the tandem dyes, which have much longer Stokes shifts, it is going to have a tremendous effect on the cores. The cores are not going to be as useful. We're only losing maybe one color in this case, but it's something we need to think about because it is something that's going to get worse and worse as the wavelengths get lower, which they will. And I'll talk briefly about the 280. It's a little bit out of order in terms of wavelength. But I bring this up because this is where the autofluorescence issue really came to a head. Oxius, which is a French laser manufacturer, briefly produced a 280 nanometer module. Very nice laser, but extremely difficult to manufacture. It was meant for the spectroscopy industry where people wanted to look at protein versus DNA. No one builds lasers for cytometers. They build them for other things, and then we use them. So very nice laser, but as you can see, if you're looking at BUV 395, it's essentially unusable because the autofluorescence just completely swamps out the signal. But one of the nice things about 280 is you do get less excitation over all of the BUV dyes, which in fact is a good thing. This data has been published. Unfortunately, the laser is not available anymore. And we really tried to characterize the autofluorescence, and you can see how strong it is in cells. This graph is there's no label here. We've simply shuttered or unshuttered the laser running PBMCs, and the amount of autofluorescence in this 380 up to about 510, 520 is so substantial that it is going to negatively impact signal to noise. We don't see a similar thing when we're running beads. So we may only be able to use the core dyes. So back to lasers we actually have. We have worked for a while now with a laser from a company called Lumini, 303.5 nanometer. So we're moving even further into the deep ultraviolet. This was a laser originally contracted by BD for DUV dye development. They dropped the project, but we were allowed to continue it. So we've had a number of these modules built. It's not very powerful. It only emits in the 20 milliwatt range. Again, this is not a mature unit at all. It requires a lot of liquid cooling. This is actually the interior of the laser. You can see the frequency doubling mechanism here. This is a 303.5, the 450 pump. You can, if you put a spectroradiometer on it, you can see the blue pump laser light. The primary wavelength is 607, and this is the second harmonic down to 303. And the fact that you're looking into this tells you that this is not a highly developed laser yet, because dust is settling into this even as we speak. Most solid state lasers now are sealed at the factory in class 100 clean rooms, and they will never be opened again in the lifetime of the laser. So this is still very much a unit that requires periodic adjustment. But it's worked well enough that we've been able to test it out. Again, this is data we've presented in the past. It excites Q dots quite nicely. And it does excite the BUV dyes, but not nearly as much as a 355, which again is a desirable characteristic for a deep ultraviolet laser that's going to be used for high dimensional flow. Now, at the time, we didn't have any deep ultraviolet dyes to test, but we could certainly run the BUV dyes in a 2-laser configuration and look at the spectral overlap, the spillover, and the spreading between a 355 nanometer aligned detector and a 303 nanometer aligned detector. They're usable together, but there's a tremendous amount of spreading. The BUV dyes just don't function terribly well in this environment. Why would they? They were never designed to do it. And also, as expected, the autofluorescence is quite tremendous. On our instrument, we can shutter the lasers on and off, and if you're running BUV 395 labeled cells, the signal is entirely swamped by autofluorescence. You start to see it recede a little bit with BUV 496, but the reality is we're only going to be able to use long Stokes-shift tandems. The autofluorescence is really quite strong. Interestingly, it sort of appears to drop off as you move into when you're into the UV on the detection side, but this is probably not real. This is probably due to the fact that the fiber optics on our instrument do not transmit ultraviolet light well at all, which is something we're trying to build our way around right now. I won't talk about label-free applications today, which are a big focus of interest in using deep ultraviolet sources, but ultimately we're going to want to have a system that can look below 370, 360 nanometer for label-free detection of proteins and other things using these systems. But so when you look at the core dyes, you can see they are detectable, but very little separation between the signal and noise. But if you look at the tandems, the DUV 530, 704, they function quite well. They're not well excited at 355, as we saw before. They are at 303, and the detection sensitivity is actually pretty comparable to BUV dyes. So it's certainly possible to build deep ultraviolet fluorochromes. We've looked at some other systems as well. There are not very many. This is a hard wavelength to build to. When Coherent has trouble, you know you're in trouble because they're very good at this. And the Coherent engineers look queasy when I talk to them about this design. This is a Coherent prototype 304. It's big. It has actually a little pressure plenum up here because the laser is sealed, but as it heats up, it expands and starts to deform the prism mounts. So they have a little rebreather up here that allows that air pressure to vent off, but it has to be isolated from the environment. Dust and moisture cannot get in. So it works just fine, but this is not a machine you're going to want to install on your cytometer yet. But it certainly does a very good job of exciting the DUV tandems. And again, the fluorescence levels are comparable to what we see with Q dots or BUV 563. And I promised you 4-color data. This is the 4-color data. Our instrument is very limited in terms of number of detectors, but the DUV and the BUV dyes are quite compatible with one another in terms of overlap and spreading doing this. So there's really no reason why we cannot combine them together. So that's where the 303 is at. We're actually building another unit now I'll talk about at the end. But can we go any lower than this? This is a laser. We actually just published a paper on this because the next stop is 266. This is it's a difficult wavelength to build, but there are a lot of them out there. It's used in manufacturing, semiconductor inspection. It's the 4th harmonic off the ND-YV04 laser. So these lasers have been around for a while, but they haven't been applicable for cytometry. They have not been continuous wave. They've been pulsed. They typically emit in the watt to kilowatt range. But there are a couple units out there that are now built for biomedical applications. Krylas in Germany and Toptika have produced a couple of units. You can see they're big. They're not very small lasers yet, unfortunately. But they work quite well. This is a mature technology. You can actually run out and buy these. They do a great job of exciting Q dots Again, having Q dots as an internal indicator to tell us what the laser, how the laser is functioning is really important. And they do a lousy job of exciting the BUV dyes, which again is a good thing. We want to see that. And if we do that same experiment where we're only running BUV dyes, but we're looking at potential overlap into a deep UV detector, we get much less spreading and much more reasonable compensation. However, the DUV dyes we have been experimenting with excite at 303. They don't excite at 266. And the 266, of course, does a lousy job of exciting them. But I've talked to the chemists, and they inform me there is really no physical chemical restriction on designing polymer dyes that excite down at this wavelength. So if there is sufficient interest and sufficient need, we can design dyes that will excite way down there. Autofluorescence, though, is still an enormous problem. It is exactly a problem the way it is at 303, and we see tremendous amounts of autofluorescence at the deep range, or in the violet up into the blue range. But again, it simply means we sacrifice our core dyes. We'll just have to stick with tandem dyes. Now, as many of you are aware, the brilliant ultraviolets are not the only UV excited dyes out there. Biorad now has a series of star brights that rely on a different assembly technology, but they have spectral properties that are very similar to the BUV dyes, and we've worked quite a bit with them. And they behave pretty much the way the BUV dyes do for the most part in terms of ultraviolet and deep ultraviolet excitation. There are a lot of other dye technologies coming along too. Some of you may be familiar with the multi-dots. These are produced by a company called Core Quantum Technologies. They're quantum nanocrystals, but they're encapsulated in a shell that is easier to conjugate to an antibody than the PEG encapsulated quantum dots that most of us have used in the past. They're extremely well excited at all the UV wavelengths. And there really is a lot of IP out there for designing fluorochromes down in this range. It's just there's not enough commercial impetus to do it yet because there are no cytometers that have these lasers. We're in the chicken and egg conundrum right now because the dye manufacturers say, well, there are no machines. The instrument manufacturers say there are no dyes. So we kind of have to break through that if we're going to take advantage. So I titled my presentation, How Low Can We Go? We can go below 266. I was just at Photonics West a few months ago, and there were a number of 261 nanometer lasers out there. This is actually an easier laser to build than the 266. The 266 relies on the fourth harmonic of 1064. It means the laser needs to be physically large. It's more complex. This is a second harmonic. It's frequency doubled off the 522 green laser diode. So in theory, this should be an easier module and a cheaper module to manufacture. I don't have one yet, but Power Technology is going to loan me one. And this is about 10 milliwatts, so not very powerful. But this is probably, if we're going to go down that far, this is probably the way we're going to go about it. Coherent has also officially announced that they're going to build a unit as well. They're building a 320 too, which they also announced this year. Not a 303, sadly, not officially. And we can go even lower than that. There's a company called Oxide in Japan that has traditionally manufactured coatings and crystals, but they recently acquired some laser IP, and they now have CW lasers from 266 down to 257, 244, and 213. These are big. They're still at the build-on-demand prototype stage. They're not very powerful. They don't have very long lifetimes, but it can be done. We can conceivably get down to the mid to low 200 nanometer range. And again, there really is no physical reason why fluorochromes cannot be built to be excited down in that wavelength range. These lasers, however, cost more than a whole cytometer does at this point. They're very expensive, so we're not quite there. I can't. But they exist. They exist, and the designs are out there for building them. More practically speaking, there's a company out in Arizona called DUVE Photonics. They are a spinoff from a laboratory at the University of Arizona. They have some deep UV technologies, and we are building a 303 with them right now that should be ready next month, I believe. So we're really trying to pursue that particular wavelength since there is dye technology out there that will respond to it. So as I've been working with these, you know, we've been trying to get as far away from 355 as we can. 320 is just too close. The two wavelengths excite the same things. So we've looked at the 305, 303 and the 266. However, spectral cytometry may throw these assumptions out the window because it may turn out that the 320 has some interesting excitation properties, not only of the UV dyes, but also all the other dyes that we're doing. So a 320 may actually be useful. It's something we're going to have to test more thoroughly. Now, we don't have a spectral cytometer that can accommodate any deep ultraviolet dye yet, but there are ways you can at least get a peep at whether what wavelength might work the best using conventional cytometry, because many of us are now doing spectral flow, and we're all quite aware now that a lot of our dyes are excited by a lot of laser wavelengths, not just the primary one. Per-CP is infamous for this. All 5 lasers excite per-CP. Even in the UV laser excites per-CP not badly at all. We need this, as we all know, we need this phenomenon. This is what provides the granularity for spectral unmixing, the distinctive spectral signature. So if we see this effect with deep ultraviolet, even if we never use a DUV fluorochrome, having additional laser wavelengths is going to be a benefit to us. I tell our people when they're thinking of buying spectral cytometers, get all 5 lasers if you can afford them, even if you're only going to do 8 to 10 color flow, because that UV laser, that violet laser, provides a lot more spectral information, even if you never actually use those fluorochromes. So we've been starting to assess this. We're only just beginning this, but we're looking at, for example, pairs of dyes that have strong spectral overlap with one another. For example, the BD570, polymer dye, and SBUV 570. These are distinguishable by spectral cytometry. Interesting, you know, which is not, they're very close. The index is pretty high, but they are usable together spectrally. But what we've been looking at, we've been using all of our lasers sequentially. We use a quantum dot internal control, and then we collect the data through the same fiber optic path, the same detector cluster, the same filter, and we look at the differences between the signal that we see with the dyes. So we're trying to assess whether we're going to get some useful spectral information out of this deep UV range. So here we're running PBMCs that are labeled with either SBUV or BV570. We excite them at 405, the quote unquote optimal wavelength. Remember, these are violet dyes now. These are not UV. But then we excite them with all our other UV sources as well. And you can see the ratio of signal changing as we move down toward 266 nanometer. It turns out totally by accident that the star bright violet dyes excite pretty well in the deep ultraviolet, better than the BV dyes, simply by accident. No one ever designed them to be that way. But we have some discrimination, some differences, both in intensity and also in ratio between the brilliant violet and the star bright violet over here. We can do, I think, somewhat meaningful comparisons by normalizing the data, because we have our violet laser, so we can normalize the signal. We can normalize the response in the UV to the excitation in the violet. We can normalize it to an internal quantum dot control, and we can normalize to both to try to eliminate inevitable differences between the signal. I mean, ultimately, what laser we pick is going to depend on the fluorochromes, their primary excitation, but this incidental, kind of accidental, if you will, excitation is also going to play a role. So what can we do with this? I think we can put a 303 on, but I'd love to see a shorter UV, and even maybe someday an even shorter one. There's a lot of room down there to work with, actually. And we can add some additional excitation wavelengths to our current array. We're not even at this point yet. But technologically, we will have units applicable for flow at these wavelengths at some point. I haven't talked at all about the label-free applications. That's a completely different talk. Honestly, that's probably a lot more interest to biologists than what I'm talking about today, because there is a lot of potential for harvesting that autofluorescence information. So I will stop there. Again, I work at the National Cancer Institute at NIH. The deep ultraviolet stuff, the deep ultraviolet dye stuff, we work closely both with Mario Roederer's group and Steve Perfetto at the BRC and NIAID, and also BD Biosciences. And I should mention all of the laser manufacturers here, both large and small, have been very generous, loaning us prototypes, sharing proprietary information. We've gotten a lot of cooperation. It's very much a multinational effort. This technology is coming from all over the place. And we owe a debt of gratitude to companies that are really willing to often share very commercially sensitive information with us. So I will stop there, and I'm happy to take any questions. Yes. Absolutely, yes. I didn't talk about, what's that? Oh, sure, absolutely. Will the UV lasers damage cells? I have every reason to believe they will, especially if you're sorting as you move down into the deep ultraviolet. You know, this has always been the question when we've sorted even at 355, whether we're damaging cells. And there is not a lot of information out there, but Spijak Darzenkiewicz looked at this issue, looking at UV excitation of cells labeled with Herx dye. Peter Lopez has done some work on this, and the indications are that yes, you probably will cause little cell damage. If you're only analyzing, who cares? If you're sorting, you might care. So, but absolutely. I'll tell you, the 266, if your sheath buffer is even a little bit dirty, it will bake the debris onto the wall of your flow cell, and suddenly your signal is dropping and you don't know why. And you look through the microscope and we have a camera on our instrument, and you can see the little wall of debris building up on the interior of the flow cell. So there's a lot of heat there that doesn't have many places to go. Even though it's a liquid stream, quartz is a terrible heat conductor, and it just bakes it on. So there's a lot of heat, there's a lot going on there. Yeah. Uh-huh. Yeah, I know, many of you know Giacomo Vacca, who's working on this at Kinetic River, and he's very interested in the deep UV for this reason. It's actually, we built a machine. It's not a lifetime instrument, but we built a conventional cytometer with him that's designed specifically for deep UV because it's something he's interested in exploring. But absolutely, absolutely. Yeah. Yes, Anna. For now, yes, for now we are. You're not really limited to it. The main problem with deep ultraviolet, if you want to look below 370 nanometers, say, is that the standard glass fibers just don't carry the signal. You have to move to fused silica fibers, which are horribly expensive. But PMTs are, if you were going to go down into deep ultraviolet signal, in theory, they can detect down to about 185. Whether that will penetrate the glass of the PMT, I'm not sure, but the cathode ought to be able to see it. I'm not sure about solid state detectors, SIPMs or avalanche, but it's something we're interested. It's something we're interested in very much, and it's one of the reasons we're building the machine with Kinetic River, because it's all air transmitted signals. It's not fiber transmitted. Yes. Yeah, we have done some work. Some of you may know Jennifer Jones at NCI. She's a radiation oncologist who looks at exosomes in the context as a biomarker for radiation treatment. And we did some work a while back with her where we attempted to use shorter wavelength lasers for scatter measurement. And scatter's hard to detect. You need extremely good beam characteristics to detect scatter. So we were sort of technically a little bit defeated by it. But I mean, in theory, I'm not sure you necessarily see smaller particles with shorter wavelengths. You'll get better resolution of the particles, perhaps. You may be able to distinguish small particle from slightly larger particle with a shorter laser wavelength. I'm not sure it's necessarily going to bring the threshold of what you can detect down. But you know, everyone's using 405s right now to look at small particles, and they're looking at 375 and 355. So sure. No, it's something we've thought about. Yes. Sure. No, very careful, very carefully. I mean, they're not visible, but you know, 355 isn't really visible either. You're relying on impact with a phosphor screen or something like that. So fortunately, ultraviolet and deep ultraviolet are pretty readily absorbed by safety goggle plastic. They don't even necessarily need a coating. So we certainly have to take that into account because as you can see on our system, it's all out in the open. I mean, we have shields we drop down in front, but the 266 Toptica, that was a 300 milliwatt unit. It arrived during the pandemic. So I was perfect because nobody was around. I was sorting for allo bone marrow transplant. I was exempted. So I was all by myself and I had time on my hands. So that was when we did that. We chased everybody out of the lab. But you know, it's really the same safety that you would use with any powerful laser, and particularly any ultraviolet laser, because you really can cause, you know, skin damage with it. Yes. Yeah, oh yeah. Mm-hmm. Yeah, you know, I think the Cytoflex has an 808 or something like that. I think yeah, the Sony has a 785 option, I think. The problem I think is going to be the fluorochromes. You know, we can excite one or two things up there, but fluorescence doesn't function the same as you move into, you know, 900, 1,000 nanometers. So our feeling on it is that it's probably useful, but you're not going to get a lot of colors out of it. With the deep ultraviolet, you could conceivably do like the brilliant ultraviolet and violet. You can have an array of 4 or 5 fluorochromes and maybe another array if you drop 40 nanometers down. So that's where we focus our attention right now. We have put near IR lasers on cytometers, but like you say, we haven't had a huge amount of interest just because there's not a lot of action out there in terms of fluorescent. Yeah, like 2. And probably that's all you're going to get. You know, the detectors aren't sensitive anymore, and fluorescence changes as a phenomena as you move further up into the near IR. But the visible space is super full right now. You know, I mean, 40, 50 colors, you know, unmixing is great, but at some point you hit a limit where it's just not practical anymore. You get too much data spreading and there's nothing you can do about that. You know, you need to move somewhere else. Yes. Oh, okay. I think it's fine to go narrower. And correct me if I'm wrong, but the BUV dyes of 10 years ago are not quite the same as the BUV dyes now, because BD and the other manufacturers go back and tweak these things. You know, they narrow the emissions, they they modify them in subtle ways to make them more compatible with each other and with other dyes. So that's true of quantum dots as well. The Q dots that you buy now from LifeTech are not the same as the ones that were released a decade and a half ago. The manufacturer goes back and modifies them a little bit to try to improve their characteristics. But I mean, the polymer dye technology, the DUV stuff's amazing because you can tune, not only to be excited at certain wavelengths, but also to not be excited at certain wavelengths. And it's really astonishing technology. It really is. Yes. You're going to have to buy a 320 because that's the only thing you can buy right now. There's nothing else. Again, there's not the dyes aren't there yet. It's really a chicken and egg thing right now because I've had talks with unnamed companies, not BD, unnamed companies, and they're quite honest. They say, look, it costs us $2 million to build a dye, so we can't. We may be able to do it, but we're not going to do it until it's needed, which is understandable. So we're trying to kind of push the laser, try to identify potential laser technology, but it's going to be slow. It's slow. It's going to be slow. Yeah. The 320 is on the ID 7000. We don't have one, but there's one at NIH, and they swear to me that the sensitivity is better on the BUV dyes with the 320 than it is with the 355. And that might be, one reason might be because of the way the system handles autofluorescence, for example. It may not be that they're necessarily better excited, but the autofluorescence might be a little, this attractive autofluorescence. So, okay. All right, thank you.
All right. Yeah. So first I want to thank everyone who's sticking it through to the bitter end. Appreciate that. It can be a long conference, but it's fun. We still do have the raffle, I promise. And we have some really good prizes this year. I do have two awards. I do have two awards to give out. We've two of the speakers. So as we do every year, when we have someone come in and give a talk, we like to give for our emerging scientists, we like to give them a little award so that they know that they're appreciated and kind of get a little taste of what it might be on the other side, or just that they'll keep coming back because we really are trying to grow a community. And so we have this year, we were fortunate enough to have people throw in submissions, abstract submissions, and we were able to pick 4 of them out of there. There's actually other ones as well we would have loved to have, but we only have so much time. And on that, I believe I have 2 awardees that are still here. But I would like to name them all right now, just for the sake of the award itself. I thought I had it right here. All right, so our emerging scientist awardees this year have been, and we saw them earlier today, is Matilda Mostrom, Elizabeth Park, Joana Bianchi, and Jayanta Mondal. And thank you guys so much for your contributions. I believe that that Jayanta is still here and Elizabeth might. I did see a little earlier. If you would come down to the podium. All right. Is Elizabeth there as well? All right. A little card. All right. So, okay. Oh, did you? Okay. Well, here. Why don't you guys stand over here? I have right here. There we go. Jayanta first, since this is the check I have in my hand, and the thank you card. Thank you so much. So we are giving our awardees. This is for those. We are giving our awardees $200 each. It's not a ton, but it might get you something. Here you go. You're shaking. Grab that with the other. There we go. Thank you so much. We always take the cake. All right, yes. Thank you. Yeah, thank you. All right, and then Elizabeth as well. Thank you so much. Like the politics, you know, politician. And please wait till at least a week to cash that. It might be no. All right. So I would also like to I'd like to take this time also to thank the committee and the organizers here. They sometimes I end up looking like the face of it, but really there's so much more stronger people behind there setting things, making sure everything's running. Like Sarah and Amber. We have people who are forced to actually sit out at the table and not be inside where all the good talks are happening. They're watching it remote, just like some of you are now. So please, if everyone would give a round of applause for our committee members and our organizers and volunteers. All right, so we do have we have a raffle of various things out there, and so we're going to first start with some of the vendor raffles, and just so you know, we do have a bag of if for anyone who wins on the vendor raffles, we will put your name back in for the money drawings from the FlowTex. I do have some great prizes as well. So first, oh yeah, is there any more cards? Or name tags if you haven't put in a card, maybe. All right. Which one are we? Got a card? Oh, that. Well, sure. Why not? We'll try it. All right. All right. And so we have a slingshot. Oh, well, no, that was my gift. That wasn't. All right. Oh, wow. This is a full out backpack, socks. Joana Gutierrez. Is Joanna here? Well, come down. I'll take it if you don't. All right. Is an umbrella and a water bottle? Match them. That's right. We can mix companies. Oh yeah. Let's mix. We gotta mix up some of these a little bit. All right. All right. So we've got we have a Miltenny umbrella and Captain T cell here. And this is Michelle Gamada Smith. Michelle, is Michelle here? All right, let's put that up. And don't worry, I'm taking the things out, but for the cash or the Visa prizes, and we are going to put them back in, so everyone will have an opportunity. Let's get a little something else in there too, and a stuffed animal. Otherwise, we'll be drawn forever. All right, so SciTech water bottle and Miss Nuranika from Miltenny. And we have Fengao. Oh, Feng. All right. Well, this water bottle is a perfect size to fit in. Yep. Another one on this one. Oh no. Aren't we? He's a bad guy. All right, and then another SciTech water bottle, and this is for Nicole Vaughn. Is Nicole Vaughn still here? Yay, Nicole! Okay, now we got BioLegend Bank. All right, so this is the BioLegend grab bag. It has this incredible puzzle that you get to put together. Some of these stickers that I actually use quite often. It has an invisible. It's got a fun little box. I don't know what that's for, but it's. Yeah. Oh, there you go. My favorite is always a little invertible. So some people don't know this, but it's actually an invertible octopus. There's the angry octopus and the happy octopus.
I imagine it's good to put on your desk so people know if they can approach you.
Exactly. All right. And so the winner on this one is Hua Chen. Ah, there we are. We want to double up on the stuffed animals on this one. All right, double up on the stuffed animals. We are gonna do, we have a Miltenny mug that we have big macrophage and Buddy. Buddy is interesting little archer here that he's a B cell. He's right on target too. And this is a big macrophage. Eating big is just too much fun. All right, and the winner for our little set here is Hung Jiang. And if I get your name, if I mispronounce your name, I really apologize. I really am trying. I really try.
The FCS Express.
All right.
It's
Other.
under there.
Oh, do we want to do the Lenko and we're going to take a break from the stuffed animals real quick. This is a big one. All right, so this is $500. So just so you know, besides giving this to Flotex, Lenko was also one of our lunch sponsors, which we very much appreciate. One of the things that we always get here is we always try to feed our people so that everyone can come and have a little breakfast and a little lunch. And it's not always the best, but it's always at least satisfying. And they were our Wednesday lunch sponsors. So if everyone will give a little quick. And this
comes with STEMI, the stem cell.
Oh, and this comes with STEMI, the stem cell, apparently, which is the real deal. And this goes to Da McKen. Sorry. And hey. All right, we have a Starbucks gift card and a fuzzy thing. This one is dendritic. Don't shoot the messenger. And then from so just so you know, this is from SciTech. Eric Barnhart pulled this out of his pocket. So he went without coffee, so one of you guys could go with it. And this is Paul Porter. There we go. Who's that guy? This is fun just because he's at the top row, just making him walk him down. You don't have to hurt yourself, Paul. All right. Can we maybe we okay.
There is
No, we still have the or yep. Oh, well, apparently we have a collector in the house. Is we have the FCS Express. All right, so yep. All right, so the next, where's the FCS Express? Okay, so we have an FCS Express and a stuffed animal. NK Stallone. So, and the winner is Kendall Friedman. Hey. All right, so just
I think they want the
so we're gonna Yep, all right. Oh yeah, the bag. Well, let's do the last one with the two stuffed animal. Oh wow, this is the Miltenny grab bag. Oh no, I need this bag for drawing though. Oh, and you have to get the bag. What's that? Do we want to do all? All right, then we can get to the cash. All right, so this one is, and you can have the bag afterwards. This is the one we have the owl. We have our little fuzzy macrophage or dendritic. Don't shoot the messenger. A pair of socks that have all these fun little things on it, and a water bottle and the bag. Oh, there you go. All right. And the winner is Natalie Jacobs. Yeah. That look, are you like, am I sure I want these stuffed animals? And just so you know, there will be a swap afterwards. So if you got the wrong stuffed animal, you can try to trade with someone else. All right. Okay. So let me get the. All right. All right. Did I? Sure I did. All right. So. Okay, and we have everyone got put back in there. Just so you know, I got 4 $50 Visa gift cards. We got two $100 American Express cards and one $200 Target card. So this is big. Do we do reverse order or do we do? Oh, you guys are mean. You guys are mean. All right, the first one is for $50. And this is Gus Wright. Oh, there you go. You can use that for parking when you come back for the ABRF local group. All right. Here's another $50 card. This is going to Sam Rowan. Is Sam still here? I might hold on to that, but no. All right. Well, you have to be. That is the one thing you do have to be here to win the cash prizes. I think it's illegal if I held it onto it for you. All right, and the winner is Natalie Jacobs. All right, Natalie, way to put that off. See, we're making sure you get your steps in today. Thank you for coming. All right. All right. The next $50 is Sadhana Agarwal. Oh, she, of course, is another one of our top row contestants. I'm going to put this here so we can kind of keep moving. All right. And our next $50, so we can kind of rotate through, am I picking any side? All right. Is Johanna Gutierrez. All right. That's lucky. It's like I said, for the cash, I always put them back in so we can. But now, obviously, once you won a cash prize, we don't go back. Still got 3 left. The next is $100. So this might be your lucky time. Nicole Vaughn.
Okay.
I'm picking the same ones on the. She's mixing them. Yeah, are they all the same names? How many? All right. Win-win. Win-win. I'm pretty sure I pronounced that. That's it. That's as good as I can do. I try. There you go. Congratulations. All right. Okay. This is the big one. And I got to make sure. We almost need to. How can we make sure to make this all fair? And the winner is Hong Jiang. Uh-oh. All right. Michelle Kamada Smith. Wow, we got double win. We had a couple double winners. Did she leave? Oh no, it's there. Oh, there she is. And she's been with us the whole time. $200 gift card. This is the big one. I get a handshake. Oh, watch out. Camera. Can't tell about me. All right. What is so I believe that's it. Is there anything else that remember? Oh yes. Okay. That's right. So let's say a couple more things. First of all, once again, let's thank our vendors for making this happen. The one reason we can make it so that this does not cost money to our attendees, where we can fly speakers in and make sure they have actually a place to stay. That's not a box in the bus station. So we really appreciate it. and we all get to eat. There is a special shout out to Kinetic River for supporting lunch today. We really appreciate that. We're really looking forward to next year. So hopefully the vendors will show back up and hopefully they got what they paid for. Another one is I'd like to is to thank our online audience. I know this is, we saw a fluctuation of everything up to one day we even had 1,000 views, which is pretty awesome. We had how many people registered this year? We had almost 800 people register, and this is both for in-person and online. So thank you, and do know that we will be having our online, put it back up, and it will go on our YouTube channel eventually. We just have to get our master technician and editor to do that, David. All right, and I'd like to thank all of you people who have sat here in these chairs. I know it can be, they're pretty comfortable, but spending 3 days in them can be a little bit much. So thank you guys. Please, a round of applause for our attendees. And yeah, so remember next year, FlowText is going to be around the same time, hopefully same place. We'll see. It's going to be here in the Med Center somewhere. And looking forward to hearing from you guys throughout the year. But other than that, look for some events that we may pop up here and there. We are looking for volunteers like usual. I am going to set up a new volunteer list mail from Google so that we can just have volunteers, because we always need someone to do something. And we're just not very good at organizing it because we're so busy. So if for some reason you wanted to volunteer before and we didn't reach back out, it's our fault. It's not because we don't like you. It's just because we're really trying to organize things and we let things slip sometimes. So once again, I'd like to say we appreciate all y'all and hope to see you next year.